Welcome all. I think I'm audible. Yeah. Can someone just give me a minute?
Yes, sir. Good morning, sir.
Yeah,
You're audible.
Yeah, good. Yeah, thank you so much. Yeah. So, during the next 2 days, today and tomorrow, we'll just go and learn something about the next generation sequencing and the systems genomics approaches. So I'm basically using the two screens. I've got some other window over here where I might really get distracted because I need to allow people. So please excuse me, if there are some small disturbances. Yeah. So. Uh, uh, this course, I think, this has really been taught several times. I apologize. I think there might be a couple of people, who have attended last week as well in the faculty development program, which I was teaching. I hope, you'll not curse me because, more or less these are the same slides, but I'll just try to, look into different perspectives and maybe, one or two new case studies, which, I'll probably discuss. I think Raju Chain would start cursing me now because this is basically the same thing which I taught some 3 days ago. Okay. So come at next generation sequencing. One of the most important challenges that people have really encountered is the big data. Okay, so if you really look into the sequencing information that is flowing across across different databases, at some point of time we may have to say, hey, look, now we are done. We need to come up with some kind of a precision based challenge. Can we really or precisely look into these particular data and say that this is what we have really done with it, and can we ask the machine to learn from it, and if at all, if in the near future, if at all, if there are such kind of sequences or mutations or some kind of variants that are really coming up, can we say that yes, these variants look something similar to this, and now we are done with it. So that is one of the important things that the next generation sequencing has really seen in terms of big data, and that's where the systems genomics, one of the fascinating disciplines in omics has really come up. So this cartoon is basically from a woman who is really lost in an island, and she writes something like help, by taking the pebble stones. But to her utter surprise, when she discovers that she's a machine learner, she's an expert in machine learning, she takes more number of pebble stones and says that yes, I know machine learning, and that is how she gets up help from several people parachuting, and even the aeroplanes come to her. That's the beauty of machine learning, which I was just trying to tell you in the first few minutes. And I think we have got a very competent team here, along with Ashwini Tirthraj Singh and my two PhD fellows, Ayam and Shalini and Ankita Kumari. We'll take you to some of the exercises, coalescing whole exome sequencing, whole genome sequencing, and RNA is taken to be precise, more towards the whole transcriptome shotgun sequencing. Tirthraj will probably focus on some of the clustering algorithms that are precisely used taking the microarray data. So as we requested you, please create an account at usegalaxy.org. Even if you have not done it, please do it. Please don't take the multiple accounts. Okay. So at the end of the day, you will be able to learn, some of the differences between the very basic entities of how to check whether or not your sequences that you have obtained from the next generation sequencing is genuinely good. Whether or not would you be able to trim the sequences, and the most important case, there are some important questions like what's the difference between the depth of the coverage and the depth of the coverage with respect to the allele frequencies with respect to DP4. And there are also small wrappers and bash strips which we'll try to tell you. And there are basically three important pipelines this particular workshop will entail. One is the RNA-seq pipeline, which is what we call it as a cuff links, cuff merge, cuff compare, and cuff diff, which I am and I will basically demonstrate. And the DE-seq, which is a well-known algorithm, which has been used, preferentially for the microarray data sets as well, and which Ashwini will really take into the case studies. Most importantly, we'll really look into some of the cases with respect to the principal component analysis. So, what if you have several data sets? How do you reach consensus? And Shalini will really look into the HR aspects of which we have been trying to benchmark. So basically, I come from Birla Institute of Scientific Research. I'm sorry for this advertisement. So ours is a not-for-profit philanthropic institute. Our current chairman is C.K. Birla. And we've got about 14 PhD fellows all over here, sorry, 7 PhD fellows and 14 scientists, and so-called 2 postdocs. We have a head R&D, Dr. Krishna Mohan, and Dr. Purnendra Ghosh, who is the director. We've got the state-of-the-art facilities. So if we really look into some of the entities, like bioinformatics, bioinformatics has really become a kind of a recipe kind of thing for every aspect. Although it was believed that it would probably lessen the scale of experimentation, the downstream subjects like systems biology, systems omics, all those kind of things have really played a huge role. The systems aspects of understanding the biology behind that. Okay, or looking into the multifaceted value where that organism thrives in a system. Okay, so for example, there might be certain conditions where we might really want to really thrive in Indian environment where we see probably 3 seasons across India altogether. Maybe if we are really asked to live in some kind of environmental conditions like Canada, where the temperatures hover around -30 or -40, we might herald in a lot of epigenetics. We may not really what you call acclimatize so fast. So there might be epigenetics associated with that. There are certain environmental changes that are associated with that. So if we as eutherians or we as eukaryotes, if we are not really able to sustain this much pressure, then the natural selection driving bacteria and viruses herald a lot of mutations. Okay, so they encompass a lot of mutations that might be rising and falling within the environment. About 1 billion of microorganisms in our body. They also adapt in our body. They also try to thrive in our environmental conditions. And these changes would drastically lead to a lot of interactions, because not necessarily a bacteria would only interact with a bacteria, but there might be interaction with respect to some other proteins as well. So all these aspects are really dealt in one important discipline called systems genomics or systems omics. But the biology behind it is nothing but systems omics or systems biology. So in a system, you always talk about major components and the minor components. So when we talk about the major components, now all those components which you really look or which you really see, from your facial entity, like the eyes, the legs, hands, and the body parts and other stuff. But if you also see, there are also some hidden components like the liver, the lungs, and all those kind of things. They might be considered as the major components, and these particular tissues are made up of a lot of cells. So there are about 1 trillion cells in our body. Each of these particular cells has got a nucleus. Okay, there are some special cases where we also have the mitochondrial DNA. In case of plants, we have got the chloroplast DNA as well, which has got its own genetic material. Each of these particular nuclei present in this particular genetic material transcribes to mRNA, and mRNA translates to protein, and that's where all those minor components in the form of biomolecules come into picture. So at some point of time, you would have some constancy in terms of the number of genes or the number of proteins at any given point of time. So whether or not that changes in the gene would really result in an optimal difference with respect to different environmental conditions or not, can be checked using something like sequencing or something what we call it as a differential expression. Okay. Nevertheless, there are certain genes that are not altered at any given point of time throughout the life cycle. Okay. So those particular genes you pretty well know, they are called the housekeeping genes. So in a systems aspect, in a systems genomics, so you have organisms constituting organs constituting organelles. Okay, you've got something similar like in vehicle. In a vehicle, you've got a minor components that are interconnected through each other using what do you call smaller components or whatever. So at any given point of time, whenever we talk about sequencing, or whenever we talk about an organism and system, there are always contexts. Okay, so a context could be defined in terms of what those particular organism entails. Okay, so first we'll just talk about ohms. Ohms are nothing but many. Okay, or money. Okay, so whenever you say genomes, the study of many number of genes are nothing but genomes. Whenever you say proteomes, the study of many number of proteins are called as proteomes. Likewise, transcriptomes, there are about 336 omices that are existing nowadays. So each of these particular organisms herald different dimensions or different contexts. Okay, those particular dimensions or contexts are nothing but the ohms which we really define. So for example, whenever you talk about plants, the plants always constitute different traits. Okay, these particular traits are associated with different environmental conditions, and these environmental conditions could at any point of time be associated with something called trait. Okay, so here although stress traits, quantitative trait loci, genes are defined as contexts, there is a chance that they might really have an interactability. They might interact with each other. Likewise, you talk about animals, you've got the feed traits, you've got the feed efficiency traits or whatever. Likewise, you talk about bacteria, whether or not it's a monoisolate or a multi-isolate, whether or not it's a pathogenic bacteria or a non-pathogenic bacteria, whether or not it will interface a host or not. All those things will look into the microbiota and all those kind of things. Whereas in humans are more complex, multi-dimensional variables, because our patterns, our behavioral patterns are not just like any of these other euthelians or other eukaryotes, because our body is pretty complex, so our phenotypes are different, and then the differential expression changes which might irradiate our body might also be variable. Okay, and that's where this epigenetics comes into picture, and that's where all these particular ohms or contexts would really change with the changing environments. Okay, so if you really look into this, at any given point of time, you've got the true positives where what in realm that particular environment at which that particular context is really making. And at any point of time, you also have got false positives, you have got true negatives and false negatives. Okay, so this is the simplest statistical representation which we always talk about that. So this was a wonderful review article which I was a part of it, and which I'm fortunate that it's highly cited. It's on multi-omics discipline as a part of my fourth postdoc in 2014 with Haji Kadamuddin's lab. So this review gives a gist of all these kind of systems genomics approach, where we may say that at any given point of time, there might be dimensions in terms of this context or omises, which I was just trying to tell you. And the most important thing is now we need to look into the number of downstream programs or downstream analysis, to check how these systems omises can really be catered into. So this is what we are just trying to learn. So this is what I was also trying to tell you. You might ask me a question, Presh. You said that there might be differential expression that might be catered. There might be something like housekeeping genes, which wherein the differential expression is not altered at any given point of time. What would happen if the differential expression count changes within the changing environment? Okay, so that also plays a very important role. So, for example, let's talk about the various phases of a cell cycle. So you've got the S phase, where the synthesis of the organism takes place at any given point of time, very rapidly in a very robust way. Okay, you've got other phases as well. But at any given point of time, you've got certain genes that might be interacting, in tandem or across, different phases of this particular cell cycle as well. So those particular things are called as spatiotemporal interactions. Okay, so the spatiotemporal interactions can be better measured using, the next generation sequencing approach, more precisely with RNA-seq, which we'll be doing, with some of the cells. Although we'll not be doing the single cell RNA-seq, but we'll just give you a kind of a gist, what single cell RNA sequencing is about. So you're jotting down to 1 trillion cells, and then you ask in your system, what is that particular number of cells which harbors more number of such nucleoid with spatiotemporal interactions. Okay, so that is where the beauty of this RNA-seq or the next generation sequencing is. So you've got about 3.2 billion base pairs of human genome. For a deployed data set, it is about 6.4. Then you jot down to 10,000 proteins per cell. Okay, and all those 10,000 proteins in a cell make up at least 1,000 pathways per cell, and that's where your differential cell count or differential expression is measured. And since this is a workshop, please feel free to stop me if I'm really going fast or this thing. So please feel free to stop me. Okay. Yeah. So that gives us that begs us a kind of a question which we were asking in the first slide. That lady where she's lost in an island. What is in store of a systems genomicist? How do we say that these particular organisms can really thrive in different conditions, and what are the impending implications for for understanding the system? So the myth here is, the problem here is, I would rather say it as a myth, not as really as a problem. The myth here is that we believe that the computational approaches would really allow us to generate different hypotheses, okay, but not necessarily whether or not we are trying to extract the experimental entity from the knowledge that we derive. We believe that we are okay with whatever analysis we are doing, with whatever small data analysis that we are trying to do it, but that's just a myth. But the real challenge is, even if you come up with a precision, even if you come up with some confounding factors, we are somehow limited by data analysis at any given point of time. Okay, and the solution here is, you need to probably ask yourself, you need to ask, yes, I've done this particular pipeline, I've used this particular pipeline, whether or not this pipeline is really giving me the results as expected or not, and whether or not this particular result is really giving me less bias when compared to the other tools. And that is how your challenges would really take up. And this is where we need to map several differential expression data, several next generation sequencing data into a compendium of high throughput expression data that is really coming up. And then we might really want to ask, yes, if you are working in one organism, whether or not this particular challenge that you have encountered with respect to your differential expression count is seen across different organisms or not. That's one of the other questions you might really want to ask. Okay. And then finally, the solution would be to look into the reference repository. Okay. You might even ask, what if there is no reference? Okay, there is something called a greedy algorithm, which we call it as a de novo based approach, where you define the entire segments of your DNA, all the A's, T's, C's, and G's, and then you ask the machine, imagining herself that all the A's, T's, C's, and G's make up a good number of genes. Okay, so this is one of the important things that you would ask. So for that, there are a good number of tools. One of the well-known tools is the Galaxy project, or the usegalaxy.org, which I was just trying to tell you. Other tools, one of the well-known tools is the Genome Space, which is largely accessed and largely funded by NIH as well. There are also some other tools which I was just trying to tell you, but they're not really rigorously used for next generation sequencing data. Okay. So the first part of my particular talk here is there is a need, to bring the challenge of understanding bioinformatics and systems genomics approach. Was at some point of time just a kind of a discipline. Okay, it's no longer. I would say it's no longer a discipline. It's certainly a kind of a multifaceted entity where you need to ask yourself whether or not you can lessen the scale of experimentations. Okay. Certain consortia, for example, I haven't really described. I was just telling about that multiomics figure. There's a well-known consortia called the Functional Annotation of Animal Genomes, from the Iowa State University. What they've done amazingly is they've only sequenced only one genome, okay, of say cattle. Okay, they haven't really sequenced, each of the various breeds of cattle or buffalo or whatever. Okay, and then they asked whether or not the genotyping by sequencing measures of various breeds of that particular cattle would really look impromptu similar to some other animal genomes that are really sequenced. Like other animal genomes could be mink, it could be goat, it could be pig, it could even be rat, it could be mice. But by and large, they're all the euthelian animal genomes. Okay. Then what they've done is, the beauty here is now they have only sequenced one genome, okay, or maybe one each genome across different organisms, and now they have the huge data sets. Now they started annotating and curating the data sets, and they might really come up with something like spatial temporal interactions as well. But to do that, to cater to the needs of these spatial temporal interactions, we need bioinformatics pipelines. And my personal interest, ever since I started my PhD in 2004 was, to look into the regulatory elements. So what if if a protein or a gene of interest is there? By chance, would there be any gene regulating that particular gene? Would there be any transcription factor influencing that gene? Would there be any long non-coding RNA or any intergenic non-coding RNAs within the segment influencing the way that particular gene works? That's where the interactions come into picture. I'll also take you to some of the case studies later. Okay, so this ends my a quick first part of my talk. So, so in the next few slides, we'll just rigorously talk about the next generation sequencing. I think we set a precedent. Why we need sequencing. Why is that the sequencing data is really important, to meet the big data challenges and all. Okay. You know the good old Sanger sequencing, right? So the well-known Sanger sequencing or the dideoxyribonucleic acid sequencing. Okay. So during those days, the erstwhile 1000 genome project where they have sequenced about 4,300 genomes, they were all, with a coverage of only 1x. Imagine, 1x is nothing but you're just, taking, one end to the other end of that particular genome. Okay. Sequencing probably once or twice from one end to the other end. Okay, and that's where you are taking that particular thing. Of course, you make amplicons, you make a copy of it, and those particular copies are sequenced. But the precision at which the depth is really measured is only 1x. Okay, so come with next generation sequencing sometime during 2007 when the next generation sequencing efforts were really launched, right from Illumina and the other players, the erstwhile the Solid, okay, and in between we also had this Enter and the long read sequencing chemistries like the PacBio, now the 10x or the Oxford Nanopore or whatever. The growing challenge at this point of time is not the depth of the coverage, but at the amount of time or the turnaround time to which this sequencing is really measured. Okay, I mean there are labs who don't mind spending monies for getting a good sequencing data by paying some more money. So it's not about the so-called precision. Okay, not necessarily the precision. I would say, of course, the precision is very important, but at which amount of time we come up with this particular data. Are we, so if I'm just giving you the sample here and post library preparation and getting the results, are they really meeting the expectations in a very fast paced way or not? That plays a very important. So moving ahead, so in next generation sequencing data, you have got a DNA strand. DNA is broken into bits and pieces, the fragments. Okay, and at any given point of time, if you have a DNA, that particular DNA is broken down into several fragments. Usually, they're all broken down to similar fragments, so that it is read for brevity in a similar in a similar way. Okay, and then you ask, what would be the case that these particular sequences that you are sequencing right from one end to the other end, these many number of mutations. Okay, so for that, what I'm just trying to tell you is, so this is a DNA strand. It is broken down into several pieces. So aftermath or premath of your library preparation, you are given the what you call during the library preparation, you are given the adapter sequences where adapters are bound like this. Okay. In case of metagenomes where you don't know the originality of the genome, several barcodes are also added like this. Okay, so the pink one here is a DNA sequence where it is read or oriented several times, and also the linkers, just like the the pug holes or the pins which you put it on the notice board, something like that. So the linkers and adapters are ligated like that, and the size of this particular DNA, okay, could anywhere between 50 bases in case of a minimum short read sequencing. The well-known sequencing player where 80% of the sequencing is done by Illumina sequencing facility is about 50 base, that's a minimum. The maximum could go up to 500 bases. That's for the short read sequencing chemistry. And usually it is oriented three ways. One is a single end chemistry where it is the read is oriented like this. Okay. In case of paired end sequencing, it is read like this and also like the reverse as well. Okay, so it is what we call it as a paired end chemistry, which is quite common to ensure okay, that it's not only scalable but also with a greater precision. Okay, and there are no false positives in case of reading that particular sequencing read. There are some labs where people are pretty rich, which I recently encountered with one of my collaborators, with the Drosophila melanogaster sequences, which I was analyzing. They used a high-end instrument called NovaSeq, okay, Illumina NovaSeq, and they used a mate pair chemistry. So in a mate pair chemistry, what happens is you have a sequence like this, okay. So each of these particular fragment is read multiple number of times from one end to the other end, likewise reverse as well. Precision, this gives you more precision. So imagine papers like this coming up, okay, and this if this were this thing, so the depth of that particular sequence can be measured impromptu whether or not the machine or the instrument has really measured properly or not, okay. And then of course they are hybridized, okay, and then after hybridization and target enrichment, the adapters and linkers are gone, and then you get the sequences, and then you can trim the sequences or the adapter sequences at the end, okay. So adapters are nothing but the pug marks, which are usually at the end, they're also just like what we usually put it on the notice board. So and again for target enrichment, the different probes, with respect to different libraries as well. So different in case of well-known library preparation. Also have got their own small molecular extraction as well. Molecular extraction is also very important. Isolate DNA from various tissues. We have had some challenges taking RNA or DNA from the FFP blocks, okay. But in case of your whole blood or in case of a normal sample, okay, maybe extracting DNA is quite easy, but extracting RNA and reverse transcribing to cDNA for your RNA-seq is quite kind of a challenging and rigorous thing. So there are different procedures. It's always advisable to keep the same molecular extraction strategy and then coalesce it with the same or similar companies in the library preparation. Although people switch extraction protocol from one company and taking the library preparation and downstream analysis with different things. Okay. So so in case of short read sequencing, the well-known approach that you could do is the Illumina short read sequencing, and the ideal size would be anywhere between 150 base pair paired in chemistry. Okay, although the erstwhile 1000 genome project heralded only the single end sequencing. There was nothing as such at that point of time. But we need to come up with a greater precision, and it all depends. If you have a 6.4 billion base pairs of a deployed genome, for a whole exome sequencing, in case of whole exome sequencing, 6.4 for a deployed data set, okay, 1.8 percent of the human genome is only the coding, okay, which means you have got for a for a haploid data set, it's about 1.8 times, something like 3, so which is about 38 MB of the exons, the coding exons for the human genome. Okay, so usually there are different library probes. There is a 38 MB capture for the in case of Agilent. There is also 54 MB. Which coalesces some of the intron exonic boundaries as well. So usually if you really take the 1.8% of the human genome, so that's ideally that comes to about 38 MB. Okay, so if you really want to capture only the 38 MB, the whole exome sequencing is only captured looking into only the exons of all the genes. Okay, and now you might really want to ask a very pertinent question. So you're only sequencing only the 38 MB. Now, what if you really wanted to know that there might be certain intergenic mutations or variants sitting within your population as well. Okay, so that's where the targeted sequencing has really come into picture. So you got the whole exome sequencing looking into only the exons, the whole transcriptome shotgun sequencing, taking the RNA as a seed material, taking the cDNA double-stranded hybridization, and chopping off the cDNAs and adding the adapters and sequencing it. Okay, that's where your differential expression count comes into picture, where you take the cDNA measures in your samples. And there's a whole genome sequencing. So imagine the entire whole genome where you walk right from one end to the other. So all the 3.2 billion base pair for a haploid set and 6.4 for a diploid set, which is pretty huge. So if you want to walk all through the genome and sequencing it, prepare a library for that, then that too, with a greater coverage, you can't do that. Okay, I mean you can do that, but it's more or less it's a very costly affair, for a short read sequencing or even for a long read sequencing chemistry as well. 1,000 genomes people have used about 1x. That was when the first human genome project was launched in 2003. Okay, and now the ideal size for a whole genome sequencing would be anywhere between 30x to 40x coverage for a whole genome sequencing, which would be ideal. So 30x times, which is 30 times the readability for your or for brevity, to check whether or not all sequences are read those many number of times. So 30x coverage times 6.4 billion base pairs for a deployed data set. So that comes to roughly 30 times 6, which is about 1.8 lakhs for a whole genome sequence for a deployed data set. Okay. So basically, the library preparation is usually done, for a haploid data set, not for the, the diploid thing. So that would reduce your size to somewhere about, 90,000 to 1 lakh rupees, something like that. Okay. And now there are more cost-effective approaches as well. For a whole exome sequencing, it's always nice because in a whole exome sequencing, it is believed that you're only talking about the mutations or the mutational spectrum seen in only the coding chunks. Okay. So if you are talking about the mutations seen in the coding chunks, then you need to be very, very sure whether or not in your given population those mutations are seen or not. So that's one of the reasons why for a whole exome sequencing, the ideal would be somewhere about 110x or 120x or 160x or something like that. But you will not cover that much depth. You would probably fall short of it. Okay. And probably you'll come down to about 100x, or even 90x, even if you have 110x, coverage. So I'll just, I'll always give you a classical example, of a person, paragliding from an outer space, coming down or jotting down to somewhere like Birla Institute of Scientific Research. Say 1 kilometer above sea level, you might see Birla Institute. But if you still come down, yes, you look that this is your lab, and if you paraglide down, then you may not really fall down exactly there, but you might really fall, somewhere in a different position. So that that thing varies a lot. That particular thing. So the erstwhile sequences, the dideoxyribonucleic acid sequences, they were all measured using something like a FASTA format, okay, which is a fast alignment format, okay, which we use it, for your blast or whatever. That's where your the normal sequencing raw reads, come from. Sequencing raw reads, are measured in terms of something like FASTQ, okay, which we call it as the fast quality files or FAST5 files in case of the ONT, okay, or in case of FAST-M in case of the iInterRent or some other instrument. Okay, so and then you get these raw reads. So now comes your question. You have sequenced your population, be it whole exome, whole genome, or whatever, or targeted or whatever. Now you want to map it to your reference. So as I told you, you've got several reference genomes that are already published. Okay. In case of human, there is a well-known build like Human Genome 38. or Human Genome 37 as well, which is a well-known build. So you take those particular raw reads and just map it to your reference. Okay. You might ask a very pertinent question again. As I told you, what if you don't have your reference? Okay, that's indeed a very good question that you should ask. So as I told you, there are some greedy based approaches. There are some de novo based approaches where the machine would learn from those particular sequences and map it to your databases like non-redundant databases or whatever. And then it's a recursive approach, where it's a greedy approach. It just gives you some kind of sequences whether or not they're sequenced properly or not. Okay, so that's where you know that's where you can do a non-reference based mapping or a de novo based mapping. 1 minute, please. I'm getting one. Uh, Sorry, I didn't give a link to so many people. I think some of them might be asking. 1 minute, please. I'm sorry. I think. So. That's where the raw reads can be mapped to a reference, and then as I told you, these particular things needs to be trimmed using cut adapt or trimomatic. Okay, there are some tools like because these are the adapters. These are only the pug marks which needs to be removed, as I told you. Okay, and this is your original sequence. Okay, so these harbors mutations. Even if you don't remove it, that's fine, because anyways, when you map it to your reference, when you map it to your when you blast it, you might, although you might get it, you'll know what these particular small chunks of adaptive sequences are, which are anywhere between 6 to 16 bases at either ends. Okay. If you're talking about the whole exome sequencing, you call the mutations, you take the SNPs, you take the mutations and check whether or not any of those particular mutations are seen in your population, not in other population. Okay, and you know the difference between a single nucleotide polymorphism and a single nucleotide variant. So the SNV is pretty unique, to your subpopulation of interest, whereas a single nucleotide polymorphism, is a kind of a mutation, that could be seen across a subset of your, sequences. And then you analyze the raw reads and you take the public or proprietary databases, like the DBSNP, Thousand Genomes, the Polyfan, SIFT, the GERB score, and other things. Then you map it. And of course, there are some pipelines, well-known pipelines. As bioinformaticists or systems biologists, we use open source tools. Okay, so the well-known open source tools are the GATK, which is a well-known pipeline for the whole genome and whole exome sequencing, where 76% of the people use the GATK, and we are among those 24%, who do not use GATK, for some reasons. And there are also some other pipelines like SecMule, OPEX, and Impact. These are the pipelines, and there are some other pipelines as well, like Taverna, Galaxy, and Tavexa. Galaxy is a well-known pipeline, which I asked you to create an account, which will be doing, the exercises. And there are also the commercial platforms, the well-known commercial platforms like the Partech, which is based in the US, the Interpretomics, which is a well-known pipeline from the Bangalore, and the Kajan CLC Bio, which is a well-known this thing. And I always keep suggesting that what matters is reaching a consensus. It's not that you really want to ask the Eureka moment or the Hurrah moment. Don't say that you have gotten the results by using only one pipeline or one tool. Okay, you may have to reach consensus. You may have to ask yourselves what is in store using this particular tool when compared to the other tool. Okay, so this needs to be done, and there are of course FASTQ files. This is your original raw rates. This is quality checked using different tools like FastQC or MultiQC. There are some tools, and then you get you map it to your reference using the sequence alignment mapping file, okay, which we call it as a SAM file. But the SAM file is pretty huge, for the bashers or for the programmers, it's always advisable to use binary alignment mapping on the BAM file. Okay? And then when you map your binaries, most importantly, if the human genome is, if the organism or the genome of your interest is very complex, you may not really get, those particular sequences in order. When I say order, it's not the chromosome wise. Okay, I'll tell you why. What you're probably doing is you are isolating your DNA, which is a kind of a mixture of admixture of a nucleoid, from the cell. Okay, you churn it, and it has got a lot of chromatin. The chromatin, when it is embedded, the entire DNA is really an admixture. Okay, so it doesn't tell you, okay, that yes, the first few strands are chromosome 1 or this thing or that thing. It will not. Okay, so you have to sort them, once you get the binaries, which we call it as a sorted BAM, and then you use something called different tools. In case of whole exome sequencing, you call the variants using various tools, which we'll be using. As I told you, in case of the GATK pipeline, you got a well-known pipeline. There are different entities. There are two different entities in case of GATK. One is something called the haplotype caller, and the second one is the unified genotyper. So you get your sequence. You imagine yourself, you ask the algorithm whether or not you've got haploid set of chromosomes all through this particular things, where you have got those haplotypes within the genome, and you want to measure the number of haplotypes or haplotype calls, in your sequencing reads. So that's where the haplotype caller comes into picture. In case of unified genotyper, the reference allele and the alternative alleles are not really tandem. They're not the same. So for example, in a reference allele, you've got C. In an alternative allele, you've got G. Okay. Then your unified genotyper will measure it as C and G only. So if there are multinucleotide polymorphisms like CCCC, and in your alternative allele it is GCCC, then unified genotyper cannot measure such kind of mutations. So there are different tools. The variant calling format is what you get the variants finally when you use different things. Then you do your downstream analysis. But at any given point of time, the most important thing is you need to tweak your tools. You need to ensure that maybe you are not running your tools with a normal pipeline and simply getting your results. Okay? There might be some instances where you might be running with some options and tweak your tools so that you can benchmark it for your previous use. Okay. So this is the quick summary of this. So you've got the parent chemistry, which I was just trying to tell you. The adapters are ligated at the end like this. Okay, and if in case of a parent chemistry, the target coverage is also very important. Okay, and usually the depth, the yield depth is very important. So as you could see here in this particular thing where my cursor is, so this defines the read depth or the mapped read depth. How many number of times, how many number of bases have been sequenced with that particular depth. So here, as you could see, this is the peak with an interquartile range of 6, where those many number of bases have been mapped with this particular depth. Given point of time, above 5 for whole exome sequencing is ideal, but even 5 is a very delimiter set because not all machines would be able to sequence those particular depth. So that's one of the reasons why you may have to look into minimum 5, which is an ideal one, and maximum is under your expectations. Recently, we had even up to 2,000, which is quite amazing to work. So 2,000 is pretty huge, which means imagine each of those particular bases, all through the length and breadth of your sequence with a 150 base pair chemistry, being read so many number of times by your machine, and then taking that. Okay. And then you may have to trim it, then you go for substitution or inversion ratios or whatever. Okay. And then usually, if you ask me, usually a pipeline, for a clinical exome sequencing, something like this, you may have to get something, once the DNA is shared, the library is prepared, the turnaround time is, somewhere between, 18 days to 30 days. But even we are getting the whole exome sequencing done in less than about 3 or 4 days. Ashwini Mishra is here. Their lab, good. So probably Ash is here. I'm not sure. So we'll get the sequencing done, with about 2 or 3 days. They got a wonderful platform with respect to Illumina HiSeq and MySeq. Okay. You might ask, what is a clinical exome? How does it differ from a whole exome sequencing? Okay. So in case of whole exome sequencing or a clinical exome sequencing, in a whole exome sequencing, as I told you, you're chopping off the entire 38 MB of the exons for that particular library capture, and you're taking it. Okay. In case of clinical exome, you know what the gene of interest is. If you're talking about a Mendelian inheritance, or if you're talking about a Mendelian genetics, or if you're talking about a monogenic diseases, okay, or monogenic disorders, which means that there's only one gene responsible for that in that particular population. So why waste anyways in the entire taking the entire whole genome sequencing? What are those particular regions of interest where these particular mutations might be heralded with. Okay, then you ask the instrument, your service provider, to look into those particular sequences only. Okay, where they're really measured. So that's where the clinical exome comes into picture. So the mutations, where they are and why they are, and then you can ask, this could be the case. So in case of exomes, you will have a lot of mutations missing from the non-coding RNAs. Okay, because we don't have the non-coding RNAs sequenced or captured. Okay. So people like me or our group are very much interested in long non-coding RNAs and other non-coding RNA players. So we are pretty much in conception that the genetic variation can be best seen in the non-coding RNAs. Okay. So at any given point of time, at any given point of intron-exonic boundaries, or even within the intergenic sites as well, there might be mutations that might be sitting between two exons like this. Imagine the pink one is an intron, and these are the exons, okay, like this. So what happens is, since the exons are pretty small, you would probably end up only looking into the mutations seen in these yellow regions. But we should also look into those regions, that are bounded by the intron and exons. This is what we call it as the intron exon boundaries. Okay, this is where you can really ask whether or not any mutations are seen, length and breadth of these particular genomes. So that's where you could ask whether or not any regulatory elements are there, any sequences that are primarily associated with different things. Yes, I hope I'm there. I think my screen is frozen. Hello, can you hear me?
Yes, sir, we can hear you.
Yes, sir.
Sorry, no, my screen was.
Yes, Prashant. Yes, Prash.
Yeah, hello, Raj. Yeah. So, and then in case of whole exome sequencing, it's pretty cheap because you're only talking about 1/7 of what the sequencing costs for a whole genome sequencing. And of course, in case of whole genome sequencing, as I told you, there's a limitation as well. The money is a big problem. Okay. And then you can identify, of course, the mutations in whole genome sequencing to the length and breadth if your lab is super rich. Okay, if not, the fallacy with whole exome sequencing is to capture the efficiency because you're not looking into the intronic boundaries. Okay, so so this is where, so finally you will come up with list of mutations in case of whole exome sequencing or whole genome sequencing as well. And then you ask, what are the benign mutations, or what are the well-defined regions, or what are the malignant mutations responsible for that. And then in case of whole exome sequencing, because you are looking more precisely, you will be able to know whether or not those mutations are disease associated or bona fide. Okay. So in case of whole exome sequencing, as of now, over 200 genes have been well defined. Okay. Because all those 200 genes harbor, different mutations through the length and breadth of the genome. One of the most important or fascinating things with whole genome sequencing and not whole exome sequencing is, in case of whole genome sequencing, you are talking about the copy number variants as well. So what are copy number variants? Copy number variants are those chunks of A's, T's, C's, and G's that are seen across different genes all through the length and breadth of your genome. Say, for example, in case of MLH1, there might be A's, C's, T's, C's, and G's, some 40 bases or some 800 bases of that particular size of a chunk. The same copy might be seen across different intergenic boundaries with some other gene, like say PMS2 or BRCA1, BRCA2, or some other genes at any given point of time. Okay, so those copy number variants are given a miss in case of whole exome sequencing. You might get it, there might be some copy number variant associated with an exonic chunk for one set of a gene that might really come impromptu, because that's a copy number variant, that are also that you're capturing as a part of your exome. Okay, so that might, be a very important thing, and that's where you can come up with a diagnostic. So this is basically the pipeline which we have also benchmarked here, which I will wonderfully explain you the pipeline. So this is a well-known cufflinks pipeline, but we have tweaked it. We have benchmarked it with a kind of 1,000 genome data, which I has done it. So what basically we'll be going through the exercise series. We'll take the human your sequence, quality check it. Then you Bouty map it. Bouty is nothing but a tool which really maps it to your reference. Okay. In some rare cases, you also take the high set mapping as well. So the difference between the high set and Bouty is, in case of high set, you'll not get those repeat regions that are masked. Okay, that's where you can take a leaf with respect to the the high set and the Bouty. And cufflinks is a tool where it tells you or it gives you a kind of the GTF files, which are the gene transcript files or gene transcript fragments actually. Okay, which means whether or not for a given size of your genome, fragments have been read with respect to per kilo basis per million reads. Okay, so 1 million is 10 lakhs. So you are taking the RNA, reverse transcribing to cDNA. So that particular cDNA is measured, so many number of times. So the accurate measure is taken in terms of 1 million basis. So at least what is that particular fragment, whether or not that particular fragment which you have sequenced is a part of this particular 1 million measure that you are really looking into. Okay, and then you get several samples. You might be having two different samples. One is a normal sample and a diseased sample. Okay, and then you want to see what is the differential expression between these two, and that's where this is how this pipeline works. And as I told you, at any given point of time, as systems genomicists or systems biologists, the downstream validation is very, very important. That really plays you, that allows you to know whether or not you got sequencing done properly or not. I'm sorry. So and then you'll get in the downstream analysis, the most important thing is, in case of RNA-seq, since you'll get the gene, okay, whether or not the differential expressions, whether or not it's upregulated or downregulated, okay, or not. So since you wanted to validate whether or not this gene exists in your sample or not, you need to run a simplest QRT-PCR and check whether or not the amplicon size is impromptu or not. Okay, whether or not you got that relative expression when you run using the PCR, that plays a very important role. Then in case of whole exome sequencing, as I told you, you run the mutations, you check the mutations, and finally you ask whether or not those particular mutations are really associated with your samples, and then you validate it using Sanger sequencing. Okay, so once you do the Sanger sequencing, you'll be able to know whether or not in real time that particular sample has got that particular mutation or not. Okay. There are also some other measures like Casper. Okay, there are also some other sequenome-based approaches where they're usually per sample per SNP based cost entities. Okay, so you take one particular SNP, you check whether or not that particular SNP per that particular sample is measured in your sample or not. Okay, so those are the well-known downstream approaches in case of whole genome, whole exome, or even targeted sequencing approaches. Okay, so this is our downloaded pipeline, which was downloaded about 26,000 times, which was well used, and I'm extremely happy that this was benchmarked as well. Narendra Meena, my graduate student who is now in Oxford, he has done this, and this is a wonderful tool. So this is basically using all the well-known pipelines. You simply give your raw reads, your normal FASTQ files, and then finally it will tell you what are those particular mutations, in your samples. This is for the whole exome sequencing. This can also be used for the whole genome sequencing as well. So basically, you take the raw reads, you quality check, you align it, and then you, as I told you, SAM to BAM mapping. You sort it, then you index it. Indexing is just like whether or not that particular ASTs, Cs and Gs are present or not, and then you M-pile up. M-pile up is once the sequencing is read with that particular depth, those many number of times, then you need to pile up them. Okay, you need to you get these particular sequences, so then you pile it up like this. Okay, and then you measure. If some sequences are like this, you may not really get it like this, so you may have to pile it up like this. Okay, and that is how you need to do that, and then you remove the adapters. I mean, removing the adapters is the first, and then you quality check it, and then you check whether or not you have got the mutations or not. Okay. So in conclusions of this particular second part, in case of the whole exome sequencing or the whole genome sequencing or the targeted genome sequencing, there are different number of tools, the next generation sequencing tools, where you can call the variants, you can call the mutations with a precise depth or a depth of a coverage. And then you can also check whether or not those particular depth of coverage is impromptu with your allele frequencies. Okay, and then you can apply rigorous, statistical based approaches. Those particular statistical based approaches, could be associated with respect to different Bayesian methods. And of course, there are variant calling approaches, different tools like SNPF, Polyfen, and Polyfen is also a database. But the well-known pipeline is BARScan. There are also some other pipelines like VT, Hanover, okay, which are well-known, pipelines. These pipelines use, something called SAM tool SEMPILE-up, which I was telling you. So, which means you have got a depth of coverage. So, which means each of those particular sequences are read, multiple number of times. And you want to make an accurate measure of those particular sequences, whether or not the mutations are seen across all through the length and breadth of those particular covered sequences. Okay, and then of course you can apply downstream bound for any correction or Fisher's test or p-value or whatever, and then you can come up with it. So the main points what we have really learned is the whole exome sequencing and the whole genome sequencing is relatively the same. The pipeline is the same. For whole transcriptome sequencing, the erstwhile microarray data sets have really paved way for the whole transcriptome sequencing. But at some point of time, you might also want to say, yes, you've got some sequences. You want to capture both, exons and as well as introns. That's where, the targeted genome sequencing comes into picture. And then, of course, you can apply the heuristics. You can go for, different 3D-based approaches, as I told you, for non-reference based approaches. Okay. And there are some things, but there might be some questions you might really want to ask. Prash, you have put it in a way that the paid-in sequencing is good, but would the paid-in sequencing handle or give an accurate capturing efficiency? Would it be able to tell you that yes, you have captured the sequences and the paid-in sequencing is the best? Okay. If you ask me, the downstream annotation, the downstream analysis is absolutely very important. And then I would say the paid-in sequencing is the most well-known sequencing-based chemistry approach, which is well-known across the length and breadth of the genomes. It's highly, highly indicative of it. So please don't worry about that. Okay. So I think we are well ahead of the schedule. So before we come up with some of the case studies, I would probably wait for a few of you to ask some questions, please.
Sir?
Yeah, please. Yeah.
Yes, sir. It was a very wonderful presentation, and within a very
Sorry, 1 minute. Yeah, I'm getting two voices. I don't know why. I'm sorry. 1 minute. Yeah.
At the customer security management support, how much in the way of resources and other support you get to actually implement this?
Oh. Yeah, please. Yeah. Sorry, Himanshu. Yeah, please.
CSAM reports and generating.
Sir, actually, I'm in my room when another meeting is going on, sir. So can I type my question?
Yeah, please, Himanshu. Yeah, no problem. Yeah.
Okay, I'm typing.
Any questions, please? Yeah. Yeah, that's an interesting question, Rosalind Sahu. Yeah, what should be the ideal ratio of disease sample and healthy sample? I mean, that's the beauty of the machine learning. Actually, you need to ask, really limited by the data size? That's again a question that you really need to ask. I mean, if you are really limited by data size, then even a five sequences would also be very good. But those 5 each sequences for differential expression count, if no such studies are really been rendered earlier, if there are such studies rendered earlier, again, that's where your machine learning, would really come into picture. Okay, so you're, I'll give you one example. When we were granted, a small project, that was a pretty small project, when I just, joined BSI here. That was for a diabetes project. We applied for a major grant, but we were given only 12 lakhs. We were obligated to stick to only 5 each samples. But then we asked whether or not such particular samples were earlier dead, because our disease phenotype of interest was pretty unique, which I'm just going to show you with the case study. Okay, so 5 Asian Indian diabetes 2, Asian Indian type 2 diabetes sample. Okay, 5 what you call unhealthy samples. Okay, they're the diabetes samples. And what is that you need to compare it with? Okay, with a similar cohort of that particular subpopulation within the same area. You're talking about the Asian Indian tag. Okay, that is a point. Or if you can compare this particular subpopulation with the already existing Asian Indian type 2 diabetes or type 2 diabetes of some other subpopulation, okay, then you can increase it to at least 8, and then jot down to jot down to 2 what you call samples. Okay, so you have a kind of a compendium. Okay, at least you can say that yes, you have got some 8 samples. Okay, that are unhealthy, which you really want to correlate with your healthy samples. Okay, so this is one of the important things where you can draw a conclusion with respect to the machine learning approaches. So yeah, Yasin asks, I do have whole genome sequencing data in a single file data format, shows it as single end, but there are 2 different barcodes. What exactly could be traumatic says error? I'm not sure, what is that thing. I mean, usually barcodes are actually, different actually. I would assume that you're talking about a metagenome sequencing for your samples. I don't know. A barcode, could be like this, okay, or it could be like this as well. There need not be a barcode, at the end, with the latest sequencing approaches, okay. So it need not be like that. So if it were a barcode, you of course you need to trim it. Even if you don't trim it as well, as I told you, you can take a measure of accurate count if you are looking into the differential express genes. But if you're talking about a whole genome, if you're really precisely looking into the mutations, it's always ideal to have a clean file like this, okay, a clean read, something like this. So I hope that answers your question, Yasin. So Imakshi asks, yeah, I mentioned about adapters or barcodes. My first question is there any signature or pattern for the adapters, or is there any chance of contamination due to the adapters? In fact, that's indeed a very good question. Adapters are well known. If you talk about the metagenomic sequence adapters, they're absolutely well known all through the length and breadth of the genome. Okay. Usually the meta genome adapters are one and the same. You simply take only one sequence like ATG, CTG, CTGA, something like that. Okay, 11 base adapter, and that is seen all through the length and breadth of the the 5' end of your sequence. Likewise, for the 3' end. Okay, but if we're talking about a metagenomic sequence, because in a metagenome sequencing, sometimes you may really want to capture, taking the measures of 16S and 18S, taking different variable regions. So for each of those variable regions, say for example, you may really want to capture only V1 to V4 or V5 to V9 or whatever. So those particular variable regions, can be captured at a different precision. So for each of these particular variable regions, you might want to have a different, barcoded sequences. Okay, so that is where you can do that. So barcoded sequences are nothing but the signature sequences. It's just like a flag which we carry like this. Yes, I'm the flag. I am the sequence. I'm representing this particular set of a fragment. That's it. It's just like a flag. And yes, there is a chance of a contamination. That contamination step should really be heralded when you have when you make the library preparation step. Okay, that's one of the reasons why the library preparation step is highly sensitive, and that's one of the reasons why the library preparation is always dependent on your sample size, sample quality. I mean, okay, whether or not the DNA is quite good, whether or not you're getting the DNA in accurate measures with respect to good excellence, good quality or not. That plays very important. And Nimisha asks a question: does pooling of samples like 10 samples before sequencing will affect sequencing accuracy in RNA-seq study? That is a very good question, Nimisha. In fact, I mean, pooling is usually done in those samples that are not monogenic, that are not complex diseases, but relatively known across all through the length and breadth. So, for example, a common flu. Okay, and then you can also talk about diabetes as well. It has also got common. Every person is diabetic. These days. Taking measure of the lifestyle diseases that we are associated with. Okay, like everyone is sitting before computer. One way or the other, the official record in India is there are about 21% of Indians that are pre-diabetic. is very alarming, So almost 1/5 of your population is diabetic. So those diabetic data sets, maybe you may really want to pool it. But pooling is done only when your biomolecule of interest is not, uh, is not impromptu, is not really good. Okay, maybe if you have 10 samples, say from 3 samples you're getting only the desired quality of DNA, but the rest of the 7 samples you're not getting it. In an ideal case, you may really want to pool it. Take the DNA from each of these 3, mix it with the other 7, and then you all mix it, and then you allocate it. Okay, and then you call them as only 2 samples. Okay, but in case of RNA, since your question is for RNA, RNA is highly labile, it's highly sensitive, it's highly ephemeral, it's usually lost with respect to the epigenetic environment that you're doing. Okay, even if you start studying RNA, maybe even if you thaw it or freeze it in the, if your sample is in a frozen or thawed and kept it inside, the lability is usually measured because you really want to see whether or not the RNA integrity is really not lost or not. So immediately convert the RNA to cDNA. Usually you might get cDNA when you have the sample of interest impromptu in real time, very effectively, and then take the sample, and then probably you may really want to pull it. Okay, cDNA pooling, getting the results for the yield is there is no link. Okay, you cannot say that you have pulled 20 samples and your yield is more. Okay, that is never the case. That is never the case. Okay, even we had this similar problem. We thought of pooling in case of our prostate cancer samples. We asked Ashwini from his experience eating, but earlier some 8 or 9 years ago we started. But that pooling was done, for some of the cat genomes and rat genomes and mice genomes, not for the human genomes. Okay. Rosalind asks, in case of clinical exome sequencing, the interested region are emphasized. Is it a good idea to take clinical exome sequencing or whole exome sequencing in case of complex disease? I think that's again indeed a very good question. I would ask, what's the nature of your sequence? That is a very important thing. Why is that you are doing exome sequencing, be it clinical exome or whole exome sequencing? Did you ever do that particular sequence earlier for such samples earlier? Okay, or whether or not it's a kind of a family-based sequencing, which means it's a part of a trio where you take the index case or a proband, and then take the father and mother samples or even the sibling as well, and then take a measure of it. Okay. You can ask yourself. So if you think that you're taking what you call for a rare disease, then I would probably ask you to go for the whole exome sequencing, taking the father and mother and probably the sibling population. That's where you call it as a family-based sequencing. If you're looking at a complex disease phenotype where the sequencing measures are known earlier for that particular complex disease earlier, and you know that the mutations for that particular disease phenotype is this, and so, and then probably you can go for the clinical exome sequencing. Okay, I hope that answers your question. So Neha asks, you said greater than 5x depth is desirable for exome sequencing. Is there a rule of thumb regarding the sequencing depth for whole metagenomic sequencing? You see, for metagenome sequencing, there are 2 different things. You don't know what that particular genome is unless until you would have gone for a 16S interpretation of your metagenome. Okay, I'll just try to give you a small example. You take water from, say, sludge or sewage or whatever. Okay, and you want to analyze the metagenomic entity from that. Okay, maybe you really want to culture those particular bacteria taken from the sludge or whatever, sewage, water, or whatever, and then do a colony PCR and check from the 16S interpretation whether or not your bacterial interest are sitting in these particular samples. Okay, that's your first point. Okay, now when you are going for when you're thinking that yes, your bacteria is not, your intended bacteria is not sitting in this particular population, in this particular sample, you go for your metagenome sequencing. In a metagenome sequencing, it's such a complex thing because you're looking into several metagenomes. Okay? And now getting a desired depth for all these metagenomes is again complex. Although you cannot say that 5x is highly desirable. Okay, so I think you would. It all depends on the library preparation for the metagenome. If you're talking about 16S, whether or not you're looking for the variable regions, whether or not you're going for the sequencing of V1 to V4 or V6 to V8 or V2 to V4 or V6 to V9 or whatever those variable regions, for that intended depth, for that particular pooling the sample, for that particular depth of coverage, then you need to look into that particular instrument as well. Okay, to meet the efficiency. For example, Illumina MySeq, would have that capability to look into, to look into the higher, coverage or whatever. Okay, and then you can go for better things. But I would slightly put it this way, Neha. Commit metagenome sequencing. I wouldn't probably look into the metagenome sequences at all. Okay, because in a metagenome you would ideally look into whether or not your sequence or your bacterial sequence is present in that and originating in that particular sample. That's the most essence actually. Okay, but if you knew from your simplest colony PCR from a 16S interpretation that this is your bacteria, okay, and then you knew that these bacteria harbors these particular genes and whether or not these particular genes are differentially expressed in your population, then probably I would go for the metatranscriptome sequencing. Okay, so in a metatranscriptome sequencing, the chance or the readability of identifying those particular gene count across the population is high. That's where you can infer upon. Okay, I hope that answers your question. So so I'm glad there are good questions posed. Any more questions, please? Yeah, most welcome. Yeah. So if there are no questions, probably I can continue. Is that fine? Yeah. So we can spend a lot of time, for hands-on or something like that. Yeah. So yeah. So I mean, let me be a bit selfish or open. We also we have done a lot of whole exomes and clinical exomes and whole genome sequencing analysis, and even the targeted genome sequencing. RNA-seq also we have done quite a lot. But for our project, especially to brush up, we thought we'd extensively discuss on the RNA-seq thing, where our PhD fellows would also take you to those things. It's also a good exercise for them. So okay, so this is one of the reasons why we are stressing on RNA-seq these days. In fact, anyways, whole genome or whole exome sequences itna ho gaya gene banks mein. I mean, there are many sequences in gene banks that you're really getting irritated looking into those particular sequencing. Okay, so this is one of the well-known case studies which we discussed. This was a part of my fourth postdoc in 2014 actually. And my boss Haja Kadamadeen, he was working with Vapen Bhagbaj on a brainchild brainchild project called Indo-Denmark Diabetes Child Project, okay, Child Obesity Project. And then what happened was there were also some other diabetes samples, that were associated. The lab is a wet lab, wet clinical and wet lab. There are a lot of rat samples, mice samples, horse, mule samples, cattle samples, okay, pig samples as well. So my postdoc, would feed pigs and cattle and rat and mice, overfeed it. His part of the project as a part of feed efficiency project is what would happen to an animal if an animal is fed, too much. Okay, And what would be the case, relative to this in relation to the Indian conditions? Okay, so what would be the case, in terms of the Indian conditions? So that was, a kind of a project, and my postdoc colleague at that point of time, she was doing a PhD. She was in the second year, Lisetta Kovalman. She found a very important biomarker called CDEC, which is a gene, and if CDEC is lower, it indicates that it's an absolute biomarker for obese population. Okay, and the other way around is, if there is a relatively higher expression, then that indicates that the people who are probably normal or who are probably of less weight wouldn't increase their weight at all. Okay, so that is how they have done a lot of stem cell based assays and different things, and with different things with obese individuals and non-obese individuals. We also checked this in humans as well. It's not a clinical trial, but we just collected the blood samples and have done some kind of an RNA-seq as well. So what basically we had was a kind of a GWAS data, the genome-wide association data, which is relatively called based on different whole genome, whole exome, and other population that is generated. Okay. So what we did was, from the GWAS data that was existing, we wanted to see whether or not the genes that are associated, that are causal to this particular population, and the mutations that these particular genes arrive with can be co-expressed. Okay, so basically you're talking about the mutations from the GWAS data present in those particular genes, whether or not they're really co-expressed. So for that, there's a well-known algorithm called the weighted gene co-expression network analysis in R. Okay, that was the data which we have taken, and we had a lot of interesting genes at that point of time. Okay, so we reached consensus and we made a kind of a simplest protein interaction network. That is what I was very good at that point of time. And then we identified among those differential express genes a lot of long non-coding RNAs, a lot of microRNAs as well associated with that. So we checked their location, the subcellular location. We also checked whether or not these particular sequences are associated in terms of closeness or betweenness. We visualized using cytoscape and then integrated this long non-coding RNA protein interaction network. And we found the CYP2C91 genotype, which is a well-known gene, that is associated with this. And this is a non-coding RNA, as you could see from the yellow ones. Okay, so all the differential expressed genes from the weighted gene co-expression network analysis, this may say we found this CYP2C91, and this is shown to be interacting with these three genes using our bioinformatics approaches. Okay, and then we got this subcellular localization as well, and made a kind of a network. Okay, a kind of a non-reference based network looking into these particular things. And this paper was published in Frontiers in Genetics at that point of time, and luckily that was reviewed by Nobel laureate David Reich, who is the director of the NEB actually. I did not know this, and then later on he contacted us, for such similar analysis, which we have done from him. And by that time, my postdoc, and we are still in touch with him. He's a wonderful, person, where we are trying to work on the long non-coding and encoding interactions. So this was the work, that Nimisha asked, what happens, if your sample size is small. This is another case study. So we had, the type 2 diabetes mellitus samples, which we call it as a sick fat. Dr. Sandeep Mathur, our collaborator at SMS Hospital, who is a well-known diabetes specialist. He has done this, and then we asked, everything, if there are some samples, I would personally ask whether or not any non-coding RNAs or long non-coding RNAs are present. Our point was to look into whether or not those long non-coding RNAs from the differential expressed data sets are associated with some phenotypic variables. Like phenotypic variables are nothing but the body mass index, the height. Okay. In case of the diabetic history, like the HOMA IR, the HOMA beta, the adiponectin. Okay, or something like that. Any of those particular samples. And then we found a very important biomarker called link01128. Diabetic marker, and this particular marker is well known, across different population. And as you could see here, um, uh, it's already published. I'm sorry, it was published 2 years ago. It's an old slide. So although we had a sample size of 60, looking into such huge phenotypic variables, even the sample size is pretty small. Okay. And we found this long intergenic non-coding RNA 01128, and we measured its expression from our sample size. So the most fascinating thing, this is probably I would say one of the first kinds of works where we knew what are the differential expressed gene counts in type 1 diabetes, type 2 diabetes samples, and obese samples. Okay, with respect to the Asian Indian type 2 diabetes. Okay, so what happens is, what Dr. Sandeep Mathur's very famous measure is that there is something called sick fat that is deposited in every humans, okay, in every organisms. Is deposited in women, okay, more relatively in what you call in these particular upper arms, and as well as thighs. Whereas men, they would also have this, but that particular sick fat is injustifiable when compared to the women. So this is what we measured. So this bundle shows only the genes responsible for type 2 diabetes. This is type 1 diabetes, and this is the obese samples, and this is the number of genes from the multimen. As you could see, there are no common genes that are within the three, but there are type 2 diabetes 176, type 1 111, and obesity is 76. And type 2 and obesity is one and the same almost. They have the desirable phenotypes. There are about 12 common genes that are differentially expressed. And of course, the most common thing is to look into the regulatory systems approach, to look into whether or not your sample size is effective, and you will find those particular samples related to your whether or not your pathways are not regulated. This is a part of these both case studies. What I've really learned during my postdoc is, the genome sequences started. There were a lot of regulatory elements. So since long non-coding RNA interaction with a protein or a gene of interest, which we have done in case of CYP2C91, gives an immense knowledge. We asked whether or not those sequences would also allow us to identify, the regulome, the regulatory interactome approach of those particular sequences. So what we did was, if we find a gene of interest, we walk through the genome upstream, okay, and see how many regulatory elements are associated. There are any open chromatin elements, enhancers, the regulons, the transcription factor binding sites, the CTCF motifs, or something like the super enhancers, or even the pRNAs, or even the long non-coding RNAs. They might have an influence on the gene, on the downstream gene, that is associated with it. So that is how we did with it. So we went into the Ensemble genome browser and we did this, and then we developed a methodology for this. So this methodology is the first classification scoring schema which we developed is for the functional annotation. There is a SNP that is functionally annotated, taking the transcription factor binding sites or shared pathways. Then a score of 1 is given. If a SNP is associated with a constrained element, then that is associated with the GERB score or present in the enriched motifs, then another score is given. And if a SNP is associated with a regulatory element, then it is 1 score is given. And that is how we developed a methodology. And this paper was also published in a good paper, in a good journal. Okay. And this is where we ask whether or not from the RNA-seq or from the CLIP-seq, the cross-linking immunoprecipitation, or from the Western blots, or from any the RNA infractome protein datasets, sequencing, any relative information we can extract. Okay. The most thing is I'm interested in long non-coding RNAs, so that is how I developed an interest. The reason why I've developed long non-coding RNA interest is I was working on hypothetical proteins, the known unknown regions, because also I've recently discovered that the hypothetical proteins are the predecessors of the long non-coding RNAs. Okay, so which means long non-coding RNAs, before they were annotated or they are annotated, they are genuinely the hypothetical proteins. Okay. So and then you might ask, Crash, why is it you are only giving some relative examples on the long non-coding RNAs, not the microRNAs? Okay. I think the answer here is very simple. The microRNAs are pretty short, whereas in long non-coding RNAs are more than 200 bases, and you wouldn't probably end up identifying those particular mutations. I mean, you would end up finding mutations in long non-coding RNAs. Okay, so that is what I was just trying to say. So in conclusions, we have prioritized SNPs and variants. That's where the 3-point classification scoring schema which we have discussed. If a SNP is associated with certain function annotation or enhanced elements or regulatory elements or the non-coding chunks, then a value is really given to that, and that is what we did that. Okay. So any questions before we move further? We are absolutely on time. If I'm going fast, please excuse me. Yeah. If there are any questions, I'll be happy to answer. Yes. So assuming that there are no questions, and I hope I'm there. Yeah. Yes. Okay. Good. Yeah. Yeah. I'm there. Yeah. I'm there. Yeah. So. Yes, Ashok. Yeah. Hypothetical proteins and non-coding RNAs. Yeah. I mean, there's no relationship. Hypothetical proteins are those particular proteins that are predicted to be expressed in an organism, but there's no evidence. Okay. But if you really look into the annotation and see whether or not that hypothetical protein is an advent of any of those 6, translational reading frames, and if we find one of those reading frames intact to be genuine, okay, and that genuine reading frame might be unannotated, and that might be coming from an intergenic chunk, or that might be coming from what do you call it, from the non-coding RNAs. That is what we have done it. We have recently published that particular work as well. Yeah. So moving further. So I'm sorry. So we'll talk about the mutations with respect to prostate cancer. So I was working on that so-called pig lab. That was where a lot of clinical data was also coming up. At that point of time, the human clinical data and all. So as I told you, my postdoc Zoe, he used to overfeed the pigs and all the animals, and then the scrotum of these male pigs it would bulge, and as a result, the diabetic pigs or the diabetic animals would also be associated with prostate. That was what we have discovered. Okay, and then that was how I developed a kind of an interest right from 2014. So we thought we would come up with it, and then when I came back to India, there were no prostate cancer studies, and then we written a small grant, which we have gotten it, from Novartis and Brain Pan, that I'm associated with it. And there's also a women scientist fellow Nidhi Shukla here, who is also doing PhD with me. She has got her own grant as well. So this particular prostate is very much interesting because we wanted to ask very pertinent questions. Because India is a diabetic capital of India, what is the percent chance that the diabetic males aged above 55 years might have an acceptable chance to be prone to the prostate cancer risk? That is the simplest bottom line question. Okay, that's because prostate is usually caused above 55 years, and of course, even the fertility rate of prostate cancer associated patients would be at risk, and that fertility measures is only would only start that particular cycle at the age of 35. So since many Indian men are getting married at the age of 33 to 39 or sometimes 45, we also wanted to ask those particular pertinent question. What is the percent chance that a person who is diabetic at the age of 53, 54, or even 35 or 40 might be prostate cancer risk? But if you really see the difference between African Americans and Asian Indians, many African Americans eat a lot of red meat. They're more prone to this thing. And Asian Indians we eat a lot of broccoli or vegetables. And yet our population so far we have done about 20 sequencing. Out of 20 whole exome sequencing, almost all of them are vegetarians, excepting one probably. And we don't know yet. The sample size is pretty small. And of course, it also depends on the low per capita. If there is a low per capita, just because people cannot really afford meat, they would only end up eating vegetables. And probably if there is a high per capita income country like US or UK, then the prostate risk is pretty high. Okay, and who knows? Genuinely, we might really be harbored with these, prostate cancer risk as well. There are a good number of biomarkers right from the diagnostics and as well as prognostics as well. So there are some kallikrein markers that are well known. That is well discovered from those particular samples taken from the digital rectal examination. And the biopsies or the necrosis, the TNM grading, okay, the tumor necrosis, metastasis grading, okay. The Gleason scores, where the Gleason score above 5, 6, and 7 are the intermediate scores. 9 and 10 is high. 6 and 7 are moderately risk, and 8 is an intermediate score, and 9 and 10 are pretty high. Okay, people have done a mallet one as a urinary biomarker, and we have done about 20, as I told you, about 110 samples or more on the anvil. We have already sent 10 more. And this is what we have done, about 10 each samples. And then we have done this particular downstream annotation, which will be going through some of the exercises. We have used the same Meena et al. Pipeline, our own pipeline, which I am and we have done this. It was their work as well as a part of the PhD. And then we asked if there are any variable grades, as I asked you, if there are any vegetarians at higher risk. And we measured the sizes and the total number of bases and the total number of reads across all those particular samples. As you could see, these are all the paid samples. Z785_1 is the reverse read. Okay, and these are all the type of samples. And we found some handful number of genes that are responsible in Indian subpopulation. Most of them are associated with mitochondrial oxidative phosphorylation, vitamin D as well, the diabetes intake and as well as the EDA, which is associated with obesity as well. And of course, the well-known the BRCA1 and BRCA2, which are the well-known mutations that are seen, and which we're also trying to work on the long non-coding RNA protein interactions as well. So this was a paper, which we published, and this was a kind of a very, interesting case, which we will do that. So the part one of this particular prostate cancer conclusions is what we observe is all patients in cohort are vegetarians, okay, and yet they are prostate victims, okay. And our pilot study revealed that they might be associated with several factors, as I told you. UST2 NP53, which is a tumor suppressor, they're also seen as a part of the whole exome sequencing mutation rate. And call 6A1 is a copy number variant. As I told you, there's a copy number variant, where you find small stretches of those particular sequences all through the length and breadth of the genome across different sequences. Okay, and of course, vitamin D and obesity risk sequences also be found. Okay, and then now we are working on something called the prostate cancer genome walking story. Okay, what we do is we take the sequences, we walk through the length and breadth of the genome of that particular area which is upstream of that particular sequence. Okay, 5' untranslated region of that particular sequence, and see if there are such guys sitting like this, the yellow ones are the enhancers. So which means if there's a mutation somewhere in the vicinity where these boxes are kept, then there's a high chance that there might be associated with high risk of prostate cancer, and they might be used for our downstream validation studies. And this work is ongoing, so we are trying to work on the long non-coding RNA and long intergenic non-coding RNA samples. And then we are still testing this and see if any of the long non-coding RNAs or proteins are associated with it. Okay. What we usually do is, you might ask a very open question, Presh, you have done a whole exome sequencing, and how come you're talking about long non-coding RNAs? So this is what the genome walking story is. You know the mutations and the genes of interest. You know what are the 5' UTR genes or the non-coding RNAs or the regulatory elements associated with that particular gene. You walk upstream and see whether or not those sequences are present. Okay, that's where you do that. And we also found a pvRNA as well, and a long non-coding RNA, which we are also looking into this. And then we identify the long intergenic non-coding RNAs and the non-exact 053810, which is under long non-coding RNA. And recently we have mixed this with our SARS-CoV-2 analysis, which is very interesting. We have developed two methods, one with Professor Keshav Singh, where we have identified something called a hijacking mechanism. I was responsible for identifying whether or not any long non-coding RNAs are present within the vicinity of the host when the SARS-CoV-2 infection host, when the COVID-2 or the virus enters the host. Usually the ACE2 receptor, the angiotensin converting enzyme 2, and the transmembrane protein receptor TMPRSS2 are present. They invite, they're just like bodyguards. They just stand like this. They invite the viral genome. But within these two genes, long non-coding RNAs, which invite this virus, and they might be transgressed. They might be deprived of their particular function. Okay, so this is what we have identified, and interestingly, these long non-coding RNAs are seen highly expressed in males in prostate. That indicates why many men are more prone to be associated with prostate risk. Okay, so we found some pvRNAs as well. They play a major role in regulation of cellular genes. Long non-coding RNA protein interactions play a very important role. They're also associated with Fasi RNA interactions, and they play a very important role for plausible markers. Okay, and the final link is what we're trying to do is the machine learning approach. Please go back to my first slide and ask why and where the machine learning is really applied. Okay, so so we have part one, the whole exome sequencing data, the part two, the long non-coding RNA data, probably we're also doing the RNA-seq data. Okay, and the part three is, can we come up with a machine learning heuristics? Okay, can we say that yes, we've got two sequences now, two sets of sequences across two measurable sets of sequences. What would be the chance that the third sequence could also have this mutation, sitting even before you start analyzing the sequences, okay, from your mutational perspective. So this is where we are just trying to do that, and I think there's the same problem and challenge which I was just trying to tell you. We're also looking into the whole exome sequencing and non-coding RNAs because there's no diagnostic landscape is at a challenge, prostate cancer. Even in India as well, we have got a pregnancy-based kit, but we don't have a a prostate cancer-like kit in India, which we wanted to develop, which we wanted to be associated with actually. We wanted to come up with this as a part of the project. It's a largely funded project. The good news is we've got some money sanctioned about out of 1 crore. We got 50 lakhs from this, and the 50 lakhs will probably come, I don't know, next year. So we'll be able to do this, and then we want to know what are those particular viably expressed long non-coding RNAs, which might be associated with that. And who knows, in the near future, in India, especially in Rajasthan, people are really getting married at an early age. Some of them actually, even the males as well. Say for example, a person who usually gets married at the age of 22 or 23. Let's assume if he's diabetic, and if he's prone to be prostate at the age of 54 or 55, then by which time unke jo pota ya whatever, ladka or whatever, might be somewhere about 23 years age, if they conceive or whatever. So what is the percent chance that this prostate cancer, if the gentleman who got married earlier, that particular mutation is transmitted to his progeny as well? That might be the case. Even in US, these hereditary prostate cancer studies have already been on annual. There's also a Nature paper as well. Okay. So which we are also trying to work towards, development of these markers, towards this. I think we are absolutely well before time, and I think I'll show you some of the exercises, and we should go ahead with that. I think we should stop here and probably some of the questions if you have, or answer some of the questions you have. So any questions, please? Yeah. So the question is, what was the algorithm name that you were mentioning about relationship between mutating gene and protein? What is a mutating gene and protein? I'm sorry. What was it I was telling? You mean the long non-coding RNA and protein? That's not an algorithm. It's an interaction method approach. It's what we call it as LinkPro or RPA Bread. It's an online-based tool. But if you're talking about the whole exome sequencing-based strategies, I think what I was telling you is about the VARScan. Okay, VARScan, BT, and ANOVA. Okay, which we'll be doing as a part of some exercises. Okay, these are the tools. Gaurav asked, does the viral mechanism have the ability to change the link properties, and also does link play any role in the defense mechanism of the host? Yeah, that's indeed a very fascinating question. I would really love to answer this. That is where this long non-coding RNA transcription approach, which we have discovered. We have communicated the paper. This is where we wanted to see. I've started experimenting this particular work in some overseas lab with my collaborator. In India, we needed some permission. We needed at least BSL-3 for this, to do that. So hopefully, we might get some kind of results towards this. But if you ask me, this is possible only when we have the meta-transcriptome data that is available. So you might want to know the differential expressed genes from both virus and as well as the host. Okay. Yeah. Yeah, please. Is there any relationship between gene responsible for diabetes and gene responsible for prostate cancer? Yeah, that's where that's a good question, Purnima. That's where we wanted to see this. As I think I've shown you some slide here. See, we cannot say that it's highly responsible. This slide, as you could see here, okay, well known. Actually, it's inducing diabetes, okay, but it is also prostate cancer risk. But whether or not it's associated in Indian population, we don't know. So this is what we are we are currently looking into. We just need more population size to do this experiment. Yeah. So any questions, please? I'm really glad that you have asked some very interesting questions. Thank you so much. I really wholeheartedly thank you. I really appreciate that. Yeah. Any more questions? This also tells us uh, that you're interested in these particular courses, workshops. Yeah. Yeah. Any questions, please? Yeah. So there are no questions. I'll probably demonstrate some of the things. Okay. If you are okay. So what I'll do is I'll probably share the screen here or here with the quality check. Okay. Maybe I need to use this. When I say this I've got another computer here, so I need to look into this 1 minute. Yes, you can hear me. Yeah. So now, you're not able to see the screen, right? I don't know why. Yeah, now I think you should be able to see the screen. Yeah. So what we'll do is, I think we'll be able to see the screen from here. Okay. So I'm just logging into my server, our server here, just to tell you that. I know how many. I don't know how many of you are what do you call? How many of you are um, bash users? But usually, for bioinformatics and all those kind of things, we need Linux-based things. Right? Sorry. Yes. So. I'll just give you some examples with respect to some of the data. I think I don't have any this thing here. Okay. Yeah. So the most important stuff that you would always talk is the most important thing is quality check. So when you get your what do you call your files now would really look like this, like FastQC files. I think I'll give you some of the FastQC files. Maybe some of the FastQC files are present in 1 minute. Usually what we do is we usually remove the sequences. Yeah. So your file with something be, something like this, it's a kind of a small example I'm giving you. It's a FASTQ file, but it's a tar file actually. It's a zip file, okay? And usually, I'm a bit surprised why there's no FASTQ file. 1 minute. FASTQ file. Sorry. Yeah. So I have a file here. Okay. So what happens is, you have GBRC underscore R1 underscore 001 dot fastq.gz. Likewise, you also have got underscore R2. So can someone tell me what is this R1 and R2? It's a parent file, isn't it? So which means you have got R1 and R2. Okay. And now the most important thing is you get which are fresh files, right from your sequencing, machine. You need to check the quality. So in order to check it, there are different what do you call programs. One is FastQC. Okay, FastQC is a fast quality check. There's also MultiQC. Okay, another algorithm. It will tell you whether or not in your sample the GC percent is good, whether or not the adapters are there properly or not, and the library preparation with respect to the average depth is quite good or not. Okay, whether or not the readability is good, whether or not the parent sequencing with respect to the 150 bases are impromptu or not. Okay, all those things you'll be able to see actually. Okay, so in this, simply run fastqc, select that particular file, copy and paste like this. Okay, and then it will see, as you could see. Can you see the screen? Okay, it is running. It says that it is analyzing and it will give you some results. Okay, I'll not waste much time. So once it is done, it will do it. You need to do both R1 and as well as R2. Okay, so I'm just stopping it. So you already, I've already gotten some results here. Okay, which I'll be able to show you here. So what I'm just going to do is I'm just copying the result file here and show you how the quality check is. Although I have the files since we have run this, I will new window like this. Okay, so RP is what you call remote copy. Okay, and I'm copying all the files that end with HTML. Okay, and of course I give the prompt like this, and then I'll get the result. So I've copied those two particular files. Okay, now these are usually present somewhere in the home folder. Since I'm also using Linux, so this is how your file looks. Okay, so this is the basic statistics. So as you could see here, the file name is R1. Okay, and the file type is convention based call, and usually it's an instrument name. And these are the total number of sequences that have been archived taking these particular sequences. Sequences flagged as poor quality. Usually by default, both percent GC is 38%. Okay. By the way, you should also know what type of sequencing data you are focusing on. Okay, that plays a very important. Okay, so which means, are you really looking into those particular sequences from the eukaryotic genome? So in a eukaryote, the normal is 52:48, which means you got 52% GC and 48% AD. That's a well-known, that's right from the good known, DNA, basis, which you have taken into account. Okay. In case of bacteria by advent of the so-called lagging of this particular intronic chunks, okay, you don't have those particular sequences all through the length and breadth, because there's always a lot of noise, because the instruments usually cannot capture what is that, and even by inherent the bacterial sequences are different. Okay, so There's a loss of noise. So if there is a loss of noise, it's indicated as N, which means the instrument cannot figure it out whether or not it is A, T, C, or G. So it usually gives N. Okay. So that's one of the reasons why in bacteria and viruses and COVID-like sequences, if the virus samples, usually you don't have the 48% GC. Okay. You might end up for such metagenomes somewhere up to 41, drastically to 33. When we have seen 29 as well. Okay, it's expected. So don't worry about that. Because that GC percent is inherent to the organisms, is inherent to the clade. Okay, but what really matters is your per base sequence quality. As you could see, this is a parent sequencing. As you could see, it's about 250 base pair, which is a 250 by 2. Okay, which means the instrument used Illumina with a 250 base pair times 2, which is a parent chemistry. Okay, and usually the adapters are here. What are those adapters? We'll see later. Okay, if all the scores are above 28, which is quite good. Okay, all the ones are in green. This is called Fred based quality score. Okay, Fred is a kind of an instrument. It's a precision scoring matrix, and then if the score is above 20, that's quite good. Okay, 20 to 28 is just moderate. 20 to 25 are just moderate, but above 25, it's absolutely good. So all the orange ones are just moderate, all the green ones are good, and all the pink ones are relatively poor. And usually the adapters are trimmed like this. Adapters are present here, okay, at the end. And as you could see, across the length, okay, you have got so many sequences until here, probably until 164, which is quite read properly. But after this, bahut ghatiya hai, it's pretty bad. It's not at all good. Okay, so maybe you may really want to trim these sequences, okay, and probably, or you may ask your service provider to resequence it. Okay, you have every right. Let me be very open. Agar you are really taken for a ride, especially the wet lab biologists in India. If this is the quality, it's a problem with either the library preparation or the machine. Okay, and library preparation, matlab the person who is really handling it. Okay, if it is really the machine, maybe the instrument is not really tabulated properly. Okay, so you have every right to do that. Don't spend extra pi towards that. Okay, I'm not sure if Ashwini is here. He will agree. Okay, that's where this is there. And then you take the per tile sequence quality, which means you have got the same 250 bases, and these many number of tiles have been accumulated all through the length and breadth of the FASTQ file that is generated. Quality score. So the quality score is this one, as I told you. This one is a quality score. So for this particular things, for the mean sequence quality is just about 20 here. The Fred score, as you could see here. But some of them, so many number of bases has got so much of average quality per read. Okay, and quality the percent A, T, C, and G, that is usually measured. So that is also measured. Usually some of them, at this particular position, the score is quite good. But for a T, as you could see at the start, it's pretty bad. GC content, as I told you, it's 38%. There's two measures. One is a theoretical measure, and what is called and the accurate measure, or the GC count per read. So as you could see, the blue one is a theoretical measure, and this one is this. Almost it's the same. It's somewhere about 38.39 or something like that. Per base N content, as I told you, there's a lot of noise. As you could see here, there might be some noise somewhere here. Okay, so as you could see, there's a big upper layer. There might be noise, so there might be a lot of N's over there. Sequence length distribution. Some of the sequences, as you could see, it's probably rigorously trimmed. It's starting with 30 here sometimes. I don't know if it is the case, which means the library is pretty bad. Okay, so and then it's elongating like this, so which means the sequence length distribution. Many of the sequences are like this until 250 bases. You've got so many of these particular sequences less than 200,000 or 200 lakh 2 lakhs. Okay, so which is less than probably, I could say, about 25,000. Sequence duplication levels, a lot of sequences that are duplicated as well here. Okay, So as you could see here, how many number of duplicated sequences and deduplicated sequences do exist. So this will also gives you a measure. But the good thing is, if you really see, not many sequences are really what you call duplicated, deduplicated, I would say. Okay, if you really look at it, it's only about 5K or more are deduplicated, which means deduplicated means number of copies actually for that particular threshold for that particular 2x250 base pair chemistry. Any overrepresented sequences, any of those particular sequences that have got noise or A's or T's all through the length and breadth, maybe there might be a lot of M's, there might be a lot of A's or T. Would they have any hit? Ye batayega. And the capture, usually as I told you, it's the adapter content. Okay, so they have used a solid small RNA adapter. So this is the adapter that they have used. There's also Illumina universal adapter. That particular sequence kya hai, aap simply Google कर सकते हो Illumina universal adapter क्या है करके, you can simply Google it and say what is that particular capture methodology. So the tiling is nothing but like this, So here what happens is this is the 250 basis for every sequence that is measured, at that particular thing. And now, as you can see, I don't know if you can see, there are some white shades over here. Okay, some of the shades here. Okay, so imagine the bundled sequence like this or stacked like this. Okay, these are just stacked like this. Okay, so when they are stacked, some of the bases that are present over here, okay, that might be patched. Each base, maybe 116 base across the tile, there might be a lot of sequences that may not really be measured properly. Okay, that is called as a patched one. So इसलिए यहाँ आपको देखिए, you can see some of them, the white patches here. I don't know if you can see here. I can see. Okay, very light, very light. Those things are maybe for that particular base, for that particular tiling. The entire thing, is not really good. Okay, so the same thing, can also be done using multi-QC as well. Okay, and then you run the pipeline. Okay, I don't know how to show you the what do you call the command line pipeline. I can show you, but I don't know how many of you are familiar with command line. Wish we could have done this with onsite, with a good, more rigorous this thing. But what we have decided is to create an account at Use Galaxy for you. Okay, so I asked you to do this. I hope everyone has got an account in Use Galaxy, please. Yeah. So in Use Galaxy, this is how your Use Galaxy is. Okay, we have been privileged to have interacted with James Taylor, who passed away this year early. He has been a wonderful person actually, and we have done. We in India and Bioclues have done the very first projects with Galaxy. I think Pranati is here and Datri. I'm not sure they are here. They have done the first Galaxy project, probably one of the foremost Galaxy projects in India. Yes. So yeah, Pranati is here. Datri is not here. They have presented last time. So what we will do is we'll take you. This is Galaxy. Galaxy is a cloud-based interface, a kind of a cloud-based server. Okay. Can someone say yes or no? How many of you have an account in Use Galaxy, please? This is an absolutely very must. I requested you 2 days ago itself. Yes, so very good. Oh, wonderful, wonderful. Oh, no, there are no no's yet. Very good. Yeah, I think this is a very sweet news. I think you can really brainstorm very well. So this is how your Galaxy is. Please don't. Oh, there's a no. I'm sorry. Sorry, Anshita. I think you should create an account and Use Galaxy. At least in the next 6 hours your account activated. Please don't create multiple Galaxy-based services. Use Galaxy.org and GalaxyProject.eu, GalaxyProject.us, and Galaxy Project AU Australia. They have got all this service. They're all 4 different servers. We're also trying to have galaxyproject.in, which Harpreet, Sundarda, and others are really working on it. Maybe we'll see if this can be hosted in Bioclues, so that's a different thing. But please don't create multiple accounts because your account will be blocked. Okay. So once you have your account, the most important, fascinating thing that you would see here is 1 minute. Yeah. So the most important thing that you would see over here is this thing, something like this. Okay. And now these you can use whatever tools, umpteen tools that you wanted to use. So simply run fastqc. When you type fastqc, it will tell you whether or not fastqc is there. So it will tell you the read quality reports. Okay, it's very easy. Let me be very open. Rather, we telling you if you do it yourselves, that would be very easy. We were never told we have done all these things ourselves. Okay, so here probably I can take some of the FASTQ files. For example, these are the files that I have. Okay, xgc575_z14_r1.fq.gz. Okay, it's a rare disease congenital pouch colon file which we have analyzed. Okay. This is a short read data from your current list. Okay. If you want a contaminant list, contamination is usually, if you really wanted, if you really wanted to check the contaminants, you can do it. You can use cut adapt or something, which I'll tell you later. Otherwise, simply ignore it. Adapter list, if you want to give adapters, you can also give a separate file and upload the adapters. Okay. And other things are not really important. Okay, and the explanation for this particular tool is given here. So this is what we have gone through that. Okay, basic statistics for this, sequence quality and all. Okay, and the reference for this. Okay. Now you have selected that and simply execute it. Okay, then it will give you a report. All the hits that you're getting in green color, I don't know why I'm saying in blue color here. Maybe the screenshot is different. If it is in orange color or pink color, it is running. If it is in red color, bright red color, which means there is an error with that particular command. Okay, so it's the simplest thing. This is the same thing which we have run through the command line. Okay, is it clear? So this is a very simplest thing, but we'll use multi-QC, which I tell you. FastQC and multi-QC, as I told you, it's always nice, to use 2 different tools and run the analysis. Okay, so so it's still running. It all depends on your speed of your connection. And we'll get the results. In the meantime, let it run. And you can. The beauty is it's a cloud. You can do multiple jobs. Okay. There is a multi-QC and map-QC. There are 2 tools. Okay. So this multi-QC is also there. And there's also map-QC as well. Okay. This is a fast QC, and the other one is, I think, some of the tools have been removed actually. Okay. Because, it's taking a lot of space. Otherwise, you can create your own, workflow as well. Okay. You can take multi-QC as well. You can run the same file again. Okay. Since it's a cloud, please be advised that in real time, the same command might be used by several number of users at any given point of time. Okay. So since several people will be using it, your job will be at stake. It might take some more time, to get it. It all depends on your speed and the connectivity issue, and as well as the cloud space and the cloud-based infrastructure your project is associated. It has got anywhere between 25,000 to 30,000 users at any given point of time, which means if I'm the user now, there are at least 24,999 users using it in real time. It takes such heavy load. Okay, so you need to be very this thing, and then you can use the multi-QC. For multi-QC, you can take what are the tools that were used. So, for example, if you use cut adapt, you can also use cut adapt and all those kind of things. There are different tools. All these tools are very good in the Proca and all those kind of things. But multi-QC has got a more rigorous algorithm. It takes different sequences. It's not that easy like what you do for fast QC. But map QC is very important. Okay. So, for example, if you know the difference between this thing, I'll tell you one. If you see fast QC versus multi QC, if you Google it something like this, then you will get the exact pie chart between fast QC and multi QC. It will have a big comparison. Okay. For brevity, I'm not going in detail, so please don't worry about multi QC at this point of time. Okay. So it's still running. It shows that several people are using. Maybe you're also running. You can also use it. If you don't have any data sets, you can go to shared data. Okay. Go to data libraries. Okay. And sets are available like AC exome. Here, if you click AC exome, there are some fast QC files available, or 1,000 genomes. We also have got some data sets. Okay, there's a Coleman, which is a well-known data set from the Iron Torrent instrument. You can also use that. Okay, you can also use Bushman analysis, which people have shared. It's a publicly available data set. You can also use it. You can also get that sample, fast QC files. Otherwise, what you need to do, you can upload those sequences like this. Here, as you could see here, where my cursor is, okay. As you could see, I'm doing here at the leftmost side. Upload your file, choose local files, or if you have an FTP, you can also do that. So choose local file, crash. I can take some of the files, something, maybe if I have some fast QC files here. Yeah, I have some fast QC files here. So I can select like this. I can take multiple files like this. You see? And simply open. And then start. Okay. So I'm not starting it because I already have occupied about this much space. I don't want to do that. This is only for your example. Okay. Only for you. Then it will upload. And then you can start, uploading your sequences. For RNA-seq pipeline, don't worry. I am Shalini, and I will take you to the other exercises as well. But we'll run a simplest whole exome sequencing pipeline during the next few minutes. Probably I'll quickly show you for the 10 minutes, and then you can do some hands-on. And then if you want to really have some quick breakup for lunch, you can go. But please return by 1:45, and then we'll have exciting session from Raj, and then also from Ashwini later. Okay, so this is more. I really wanted all of you to do a lot of exercise space. This is very important. Okay. So so it's, you see, it's still running, as you could see at the extreme. I don't know why it is this much. Sometimes it's always a good idea to refresh. Okay, so and then based on the quality check, you can see. I'll show you how to visualize. I don't know why. Usually it doesn't take this much time. Usually they data in a matter of 4 minutes. That's an ideal precision time for about 10 Mbps speed you have. Okay, I don't know why it's taking this much time. Maybe several people are using it, and sometimes the local IP. If several people are using it with the same thing, maybe Shalini, I am, others are also, Nidhi are also working on it. Okay. So this is the case. Now, in the meantime, the most important thing is, as I told you about, something called mapping the, mapping your FASTQ files to your reference. Okay, you must know what the reference is. The references are already built up in the Galaxy server, so please don't worry. Most of the references, since there are some client genesis like Samira and Yasin and others. I don't know. I've experienced some kind of a problem with the plant genomes recently. I was working on Lycopersic on the tomato genome. It's not really good. Yeah. So for 1.5 GB for 10 Mbps connection, 1.5 GB times 10 Mbps connection, it should take less than 15 minutes. Okay. So like 1.5 times 10, times those number of bytes that you are analyzing. Okay, that's how the calculation is. If it is taking 4 hours, then that's pretty bad. I think maybe your internet connection is pretty bad. Maybe it's not 10 Mbps. Maybe the outer bandwidth is showing you as 10 Mbps, but it might be only 1 Mbps or something. Okay. So where were we? Yeah. So that's where we have the fast query, and then you need to map it to the reference. So first, there are different tools like Bauti, HiSat, Burroughs Wheel Aligner. Okay. There are also some other tools, which are something similar to that, which I'll just tell you the difference between these tools. So what happens is whenever you have your FASTQ files, you need to map it to your reference. Okay. So that mappability is very important, because if you're reading your sequences, your quality check, be it the 150 base pair chemistry or whatever, those particular fragments. Okay. Imagine this is so imagine this is your raw reads. Okay, this is your reference. So what happens is it matches like this. So there might be matches like this as well. So which means, okay, some of the sequences are matching from middle till here. Okay, and if you really split it like this, so let's imagine you have sequences like this. So you have several things like this. So these are also like this. So you might only map several reads to only one region. Oh, my video is off. No. Is my video off? Oh no. No, I think my video is on. No, I can see my video is on, but I don't see my face. I'm sorry. I don't know why. Yeah, it's because I'm probably sharing the other screen. I think my video is on. Yeah. Don't worry. So this is how your sequences are. Okay. So now the most important thing is you need to map to your reference. So there are different algorithms. One is the Bauti 2. Okay. There are also some other algorithms like Burroughs Wheel Aligner. Okay. And it's their accessory tools which have really come with other tools only. That the model is from the same Trepner lab. Okay, so the most well known is the Bauti 2, which is a readable reference. If you click here, okay, so here you need to take this. You are taking this sequence FASTQ file. If it is a single end, you need to give like this. But it's a paired end, you need to take paired end rate. Okay. You see multi-paired Galaxy has removed it because it will take a lot of time and it will take a lot of memory as well. Okay. So you can take the same file XGC575 Z12R1. Here you can take Z2, Z12R2. Okay. And you can also have an option like write unaligned reads to separate files. If you want, you can click yes, okay, or no. Okay. Write aligned reads to separate files. This is where we have seen in the multi-QC example. Okay, I've shown. Do you want to set paired end options? So yes, you want to have some paired end option like fragment length for varied paired end rates and all this. These are fragment orientations, which I'll tell you later. It's just like forward reverse strand, which means for a paired end, it's a forward and reverse strand, and reverse forward strand. First one is reverse and then forward strand. Or take only the forward reads, because reverse reads are not read anyways. Okay, that is an option, you can do that. And disable no mixed behavior. It's whenever you keep it as that particular option, yes, then there is no concordance, which means there are 4 pieces here, as I could see here. Okay, when it is mapped to the reference, okay, it will not take the consensus of all these 4. It will only take the one like this. And Dovetail is a well-known what you call library preparation thing. It will tell you whether or not it has got the exact sequence impromptu or not. And disallows mate alignments. Mate alignment is nothing but a mate page sequence or something like this, which I was telling you. So go with the default option. Don't worry about all those options because we don't have this much time within 2 days to tell you each of these. So don't go with this. So you can say no here and use default, save the body results. If you want email notification, you can keep email notification as well or no. Simply run execute. Sorry, I think I didn't take the reference. I'm sorry, 1 minute. It's a built-in genome index. Okay. And then I'm sorry, by default it's mapping to baboon. So you have to take Homo sapiens, type Homo sapiens. So there are different things, AG38 and AG17, AG37 and other things. So the build is AG38, there's a 36 as well, okay. AG18 or AG38 is here. Yeah, sorry, AG38 is here. Okay. This is a well-known thing. That's the latest one, which you can see. Okay. Other canonical, other mitochondrial sequences are also there, and then you run it. So you can probably. I think I've taken only. Sorry, I've taken only read one or read two again. I'm sorry, I've taken only read one. I can cancel those two jobs. This R1 and R2, and take Homo sapiens AG38. Okay, and run the command, and simply execute it. You can cancel these jobs. You don't need it because I've given a wrong job. So then it will map it to your reference. Okay. It will take some time. As I told you, it all depends on this thing. And then taking those things, so what is the file that you obtain? It's called SAM file, the sequence alignment mapping file. Okay. But that's not the file that you need to use. You need to convert it to BAM just to save a lot of space. You see, I got fast QC results now, so I can visualize fast QC. This is view data. Okay, when we click view data, report. You see the end reads. This is a box plot. The end box plot. Some of these things are just okay. It's expected. You can leave it. You need not give it an excuse because these are all the adapters. Adapters anyways. Could be trimmed. Okay, if you want, you can trim it. So for trimming, as I told you, fast QC ke baad, you can use cut adapt. Okay, or trimomatic. Okay, it's cut adapt is remove adapter sequences from the FASTQ files. So it's usually preferred for both exomes and transcriptomes for all the NGS, but we have never been interested in that. So you can take this, take the FASTQ file, or if you want to take the paid end or whatever, you can do it. For brevity, I'm using single end file. And if you want any frontline adapters of your own interest, you can use it. Okay, you can write typing, simply execute it. Okay, and then it will remove the adapters. So you might want to ask a question. So when you remove the adapters, so what happens? This is your sequence, and this is how it is. The adapters are trimmed. Original sequences. You will get the genuine sequences. Okay, and it's still running. Okay, so these are the two commands, and then after that, I'll quickly tell you how to run the SAM tools and the other thing. Okay, and then I think you should have at least 1 hour of exercises from 12:45 to 2 o'clock. Okay, if you have 15 minutes lunch, at least you should do that. It will take some time. I'm really sorry. The internet is pretty bad today. Yeah. So in the meantime, if you have any questions, I'll be happy to answer. Let it run. Unless and until that runs, I cannot move forward. Yes, please. Yeah. Yeah, please, if you have any questions, I'll be happy to answer. So, yes, certainly. I think, if you really want to go back and see from the traditional FASTA files, the FRED score is very, very important. There's something called FRED FRAP scores. Okay, FRED is a base quality. The FRAP is an additional quality and measure. Just like p value and q value. You check the you check the measures. Okay, so I think it's very, very important. Yeah. No, there's no shortcut, Yasin. I'll tell you one thing. Then you should owe me a chocolate. Okay. The thing is, you start the upload when you go to the lab, when you leave the lab. Okay. In the night, especially, if you are there, and upload it, and ensure that you open what you call a private window, not a normal window in the browser if you're using it in Galaxy. Okay. What happens is, the normal browser is added with a lot of, things. It loads a lot of memory. And please don't use the Chrome browser. Please use the Firefox. Chrome, although people are very big fans of it, it's not really good. Okay. It's the lightest browser, but the lightest browser will take a lot of weight. And by the time in the morning, if you come, your files will be uploaded. And don't think about that. Once you start uploading it, don't think about that. Let it download. If you keep thinking that it's very slow, then probably you'll not end up uploading them. Okay, so that's this thing. I think it's the results are coming up now. Hopefully, we'll see. And the cut is also pretty slow. Yeah. Yeah. Sorry. Oh, thank you. 1 minute. Sorry, thank you. बताइए अच्छा शादी
हाँ वो बच्चे की शादी थी ना
सर 1 minute.
जी सर थैंक यू
So I think I'm getting these results. Yeah. So what is the sequence duplication levels and overrepresented sequences represent? Yeah. If you have really looked into that, I think I've shown you those files, these things. Okay. So this one. Okay. So as I told you, this is the cutoff. Okay. So these are all those particular sequences that are deduplicated, which means that are not duplicated given the total number of sequence counts that you have. Okay, so it's following the same path. Okay, and overrepresented sequences are those sequences that have long stretches of A's or T's or G's or C's. Usually A's and D's are there because these are the sequences that constitute the adapters. N is a noise, which means it's not read. Okay. So either it couldn't read whether or not it's A, T, or G, or C. Okay. So I think it's still running. I'm sorry. So in the meantime, you can do all these particular exercises. I'll just tell you as I do it. Okay. Otherwise, I'll just show you now. So next comes the SAM tools. Okay. So SAM tools are nothing but those particular tools where you can filter your sequences. So you map your sequences, right? So which means your bounty aligned sequences, okay, that are aligned to your reference would have a file called SAM file, okay, sequence alignment mapping. So that should be converted to BAM, okay. So which means the bounty results should be taken as an input here, okay. And the output format is BAM, or you can also take it as CRAM file. CRAM is another format where you can convert between various alignment sequences. Okay, CRAM is convert read alignment mapping files. Okay, which we call it as CRAM. Okay, so BAM is binary alignment mapping. SAM is sequence alignment mapping. BAM is better. CRAM is less sensitive when compared to the other things. Okay, it takes the same input as your SAM and then it does it and then execute it. I've taken it from my previous history. Okay, so you should follow the same history. If unnecessary history is there, please delete it. Okay, you will get BAM, and then you can go for something called SAM tools sort. Okay, in SAM tools sort, you'll get the BAM file output and then take it and simply execute it. Okay, then you'll get all the things in in what you call in chromosomal position. Okay, in order, chromosome 1, 2, 3, 4, and 5, which I'll probably show you here. So I requested Yasin for a chocolate, then I got a sweet here. So first from someone, thank you. So you get the results, it's very slow. So I've done this, I'll probably show you some of the results of sorted BAM, which I've run earlier. Yeah, this is sometimes sort, okay? So when I see view data, you see, this is how your file is, its header, okay, like this. Header version number, VN is version number is 1, okay, it's a coordinate number. Sequence, serial number, chromosome 1, and that particular sub-accession number, okay. This is how it is, right? So you see all the chromosome 1, say like a chromosome 2, 3, 4. I've taken a small file, so that's the reason why I'm getting very good results, okay. We are taking this, okay, and X and Y and unknown as well. See, there's also mitochondrial sequence as well. Okay, this is how it is. In a normal sort file, you don't get this sorted order. And you can also mpileup it, because in mpileup, if you have several sequences that are sorted, okay, then you can also run SAM tools mpileup. That's an optional one. Okay, and then you got a sorted BAM, and now you can go for a variant calling in case of whole exome or whole genome sequence. Okay. So for variant calling, there is if you simply type variant calling, there are plethora of tools. Okay, and of course, the well-known variant calling is VerScan. Okay, but there are also other tools like ANOVA as well. So you can also use VerScan. Okay, and you can run your VerScan on any of these things. Okay, there are also other things: minimum read depth, minimum support reads, minimum base quality, minimum variant allele frequency. It all depends on the type of phenotype that you're working on. Okay, so if your phenotype is by and large bigger, well known, like diabetes, then you might really have a minimum allele frequency of 0.05. Okay, usually across the subpopulation, 5% of per chance that the alleles might really vary. That's a cutoff. For a rare disease, 1%, which means it's a rare disease that that particular allele frequency would really match with 0.01. Okay, so that's where you can really look into that. Otherwise, simply take all the default parameters and simply execute it. But if it is a reference allele, then you can sorry, if it is a rare disease, then you have to take 0.01. Okay, then you'll get the pipeline. And the final VCF is obtained. Okay, which I'll show you here. So this is a VCF file, you see. In a VCF file, you've got a chromosome, you've got a position, you've got a reference, alternative allele, quality, as I told you. Okay, Quality is nothing but, at any given point of time, the quality of your file, which is a measure of above 20 in terms of that. Okay. There are two qualities. One quality is taken from the Illumina instrument. The other is a kind of a friend-based quality, but this is a kind of a cumulative. Okay. And the file name sorted BAM, whether or not it's heterozygous. This file I've really uploaded. Heterozygosity is a measure where you will have different alleles sitting across two different entities, and the depth above, as I told you, above 5 is always good. Can filter it. What is a forward reference allele and alternative? Yeah, this I've measured it. This is my own VCF file, which I've told you, which I'll tell you maybe in the evening. Okay, and then finally the allele frequencies and all those things, the minor allele frequencies. Okay, so this is how your normal VCF files look. I'll tell you some other VCF file maybe somewhere here. 149. There are some VCF files. Yeah, in fact I removed my own VCF files a lot, but this is how your VCF file looks like. Okay, this all more or less is the same thing. I'll show you the VCF file from my server. So yeah, so sorry. Don't worry about this. These are the other commands which I can't tell you now. Yeah, so I type head 200, something like this. So the first 200 lines of are obtained. So this is how it is. So it tells the file format VCF4 version, the header files, and it reads all the contig lengths and all those kind of things. If you go down, I think this is how it is. Okay. So the headers I'll just show you. 1 minute. This is all chromosome 1. Yeah. So it's a chromosome, position, ID, reference, alternative allele. Okay, you see in case of reference, you see here you have a multinucleotide polymorphism allele with CAGN and alternative is CA, GA, GA. Okay. Quality, filter, info. In info you have the depth. Okay. And you have got the VDB and other scores, variant database file format. Okay. The other sequence, other things, and this is the heterozygosity, also the homozygosity. Okay, this is 0 by 1 is heterozygosity, and then you will get the quality scores and the file and all. So this is how it is. Then this needs to be converted. So which means for chromosome 1 at this position, you have got a reference called at this, and the alternative allele is this, and the quality is this. Okay. So the quality above 5 you can filter, or maybe since the quality is above 20 for most of them, then you have to map these particular VCF to your proprietary databases that are publicly available, which probably I'll tell you later. I think this is too much. I think we're time. At least I thought I should give you time for 1 hour, for you to do exercises. Okay, I'm available here. Please unmute your microphone at any point of time. Okay. And just ask my questions, but please do these exercises for the next 1 hour. Okay. Yeah, please just leave me what you call questions, if you have. In the meantime, if these results are out, I'll just show you. Okay. But I think the results are not really out now. Okay. So any questions, please? Yeah. I hope you're all doing the exercises. Maybe now we would like you to split into groups if you're interested. So assuming there are no questions, so please carry on. We'll meet at 1:45. Okay, in about 40 minutes. Huh? Please. If you have any questions, please type in. Yeah. Thank you.
Hello. Hello, Pat. Hello. Prakash.
Yeah, any questions, please?
Hello, hi Prash.
Yeah, please, yeah.
Okay, I just ran samples sort. It's going on. Okay,
Yeah.
after this, I need to do index, right? What is the command I need to use for index?
You ran quality check?
Quality check is done. First QC I have done. Then I run cut
Yeah.
adapt, and then I ran bow tie. Okay.
Yeah.
Then SAM tools I ran for conversion.
Right.
Okay. Now SAM tools sort is running.
Right. So the most important thing is, once you obtain the BAM, you're trying to use something like sort, which is, your BAM file will be sorted now.
Right.
Okay. And if you want index, that's a kind of an optional one. The indexing usually is not really done, which I'm just going to show you, when we all meet at 1:45. Okay.
Okay.
So for an online, Galaxy-based thing, the indexing is already done, by default actually.
Oh,
Okay.
I see.
But if you want to index it, you can use SAM tools index and then get an index, for your, BAM file. Okay.
Okay.
If not, you can directly go for the variant calling approach. Okay.
Okay.
And call the variants directly. Yeah.
Okay. Okay. Then after SAM tools, I will try to do variant calling.
Yeah. Please, sir.
Yeah. And after variant calling, the further steps you will be showing after 145 or?
No, it's done actually. Basically, you get the variant. After the variant calling, you need to map it to your, databases, which I'll probably, show you, once everyone is really done with this, maybe in the evening or maybe tomorrow morning, I'll show you.
Okay.
How to do that. Yeah.
Okay.
Let's meet in about 25 minutes. Huh? Thank you.
Yeah. Thank you. Hi, Praj. Hi, Praj.
Please, one minute, huh? Yeah, Savud, yeah.
Yeah, thank you, Praj. Actually, I'm able to run variant scan and able to get a VCF file.
Yeah.
Okay, in this VCF file, what are the particular things I need to see for any deviations or variations?
Yeah,
in
I
this
mean,
file?
can you please just share your screen so that I can,
Okay,
yeah,
sure.
maybe I need to stop sharing my screen here. I don't know if you can do
Okay.
that. Yeah.
Are you able to see my screen?
Yes, yes, yeah, yes, Savud,
Okay.
yeah.
Yeah, this is the VCF file. I got it.
Yeah, I think, it's pretty small. Yeah. So can you please zoom in? I'm sorry, Yeah.
Oh, is
Yeah.
it? Sorry.
Yeah. Good. Yeah. So if you can just come down, yeah. If you can visualize it, yeah. No, the first column is chromosome. Okay, it's the same thing.
Okay.
Can you tell me what the background data about your files? Yeah.
Okay. I got a 2019 coronavirus
Right. Yeah.
accession number, and then I
Yeah.
used the HG38 as the reference genome.
Okay. So these are the human samples, right?
Right. On human samples, I'm
Okay. Yeah.
trying to validate coronavirus.
Yeah. So you mean it's a metagenomics sample, is it?
Uh, Actually, in the input, when I was doing quality check, right, I used input first Q file as a coronavirus first Q file.
Yeah.
Okay, and then I used human genome as a reference genome. Just want to see like how much it is aligned with the human genome.
Yeah. I mean, no. Savud, there are two things. No, if you're
Okay,
talking about a coronavirus file, it's
okay,
a viral sequence, isn't it?
right.
So you need to probably map it to a viral reference,
Oh, is it?
okay?
Okay,
Because you are really looking into the viral genome, whether or not though the inherent virus that is really causative, for the
okay,
sample that you have isolated has got any mutations.
okay,
Okay, so if it were a metagenome, when I say metagenome, and you don't know the sequence, you don't know the virus or bacteria or whatever,
okay,
then probably your pipeline is something different.
okay.
Okay, so for this, since you're going for a viral assembly, this thing, this is not a pipeline for you to analyze.
Oh, is it?
Okay,
Okay,
yeah. So for eukaryotes, especially, if you're looking for mutations, if you're looking for inherent changes in your eukaryotic sequences,
okay.
then only you would run this particular pipeline, which I've shown you for whole exome sequencing.
Oh, I see. Okay,
Okay. So for this, the pipeline is different. Unfortunately, we'll not be discussing the metagenome or the viral assembly here. No. Yeah.
okay.
But what you could do is, instead of the human genome as a reference, you have to give SARS-CoV-2 as a reference. Okay.
SARS-CoV-2 is it? Okay,
Yeah. And then map it, and
okay,
then you will get. Then of course the procedure is the same, but you'll not be using the VAR scan for that, because you'll not be finding the inherent mutations, like what you see in the eukaryotes, but you'll find, across the length and breadth any changes, in the given sequences.
okay,
Okay.
okay,
Yeah. So don't use the human reference, for a viral genome.
okay, okay,
Yeah. That's it.
okay. Yeah, thank you.
So welcome back. Do you have any questions, please? We have Tedraj coming up. So in the meantime, we'll discuss, some of the things which I thought of discussing with you. Yeah. Yeah. So if you have any questions, please. Yeah. There were a couple of points, that we discussed. Sahud, he asked, some questions which I thought of discussing with you. So, Digden, do you have any questions, please? Disha, Esther Jamir, Gaurang, Himani, Jyoti, Kikrusenyo, Yasin, Sawad, Madhumita, Nishu, Pratibha, Rajal, Rashmi, Rosaline, Vineet. Yeah. Do you have any questions, please? Yeah. So, if there are no questions, probably I'll just show you our pipeline as well. The whole exome sequencing pipeline, which we have, used to infer these sequences. Okay. So probably I'll share my screen again. So, so usually this is how the VCF file is. So we ran the whole exome sequencing pipeline, right? What happens is we write a kind of a batch command, for everything. Okay. So this particular contains all the sequences which I'm going to show you. Yeah. So I'm not sure if you're able to see it. I'm just zooming in. Okay. I'm not able to zoom in properly, but yeah. So what happens is this is how your script is. So first, you index your raw reads with respect to Bouty, Baroville, Linus, and SAM tools. Okay. So this is your reference. This is my path, which I've given. And then, so what I use, what we did was Bouty2 minus X. Okay, is an option that is given, okay, indicating that your reference genome is here in this particular path, and -1 is the forward read. This is a forward read I've given, and -2 is the reverse read. Okay, and the output is obtained with a file option called -s. -s is give me the SAM file. So which means your raw reads R1, R2 is entailed with a SAM. Okay, and then you import your SAM tools. Okay, and then this is your reference. Actually, you need to build a Bouty reference. Okay, a Bouty reference is nothing but Bouty build is an option where your reference genome would be available as per the Bouty mapping file. Okay, so that thing probably I can give you a simplest example using Bouty build. Okay, something like this. So here and a reference name. Okay, so when you type Bouty2 build, okay, and a reference name, say for example home ng like this. Okay. So this is how you give your reference file, okay? And you give your reference index. What is the name, that should be carried out like this, okay? So which means you're trying to make a kind of a built-in taking the Bauti configuration. So when you type it, you will get your Bauti reference. All the entire human genome 38, is built up. Okay, I'll not show you because it will take some time. I'm just canceling it. So the output of this particular file is seen here in this particular path. Okay, so you'll get something like this. You see, hg38.1.bt2, hg38.2.bt2, 3, 4, and reverse 1, reverse 2. So the entire human genome 38 of 3.2 genome for a haploid set is split into these particular number of files. Okay, so this is what we meant by the Bauti build. Okay, and then this is what you need to give. Of course, this is FNA file, and this is what your Bauti build. Okay, and then you are importing those files, and then you are converting your SAM to BAM like this. Okay, and then what you do, you sort your BAM. .bam. This is output greater than symbol. That's how the output is rendered. And you can index, as I told you, index is an option, but it's nice always to have an index option. That is what Saud was asking. Okay, so you can take your BAM and then you'll get a BAM index. It's just like any kind of an index reference that you'll be getting it. Okay, and then many a time when you index it or when you map it and when you merge it or pile up, you'll get a lot of files. You can sort the number of sorted files into one. Okay, this it depends on the type of the file and the size of the file. Okay, and this is how you get it. And SAM tools mpile up. Okay, SAM tools mpile up sorted. So you will getting all the mpile up sort. There are mpile up 1, 2, 3, 4, 5. So many files that are done, and that would be mpile up dot bam. Then you run the var scan for the pile up. Then you'll get SNPs. You'll get indels. SNPs and indels, the difference, right? So the SNPs and indels are something different. SNPs are the single nucleotide polymorphisms, whereas indels, there is a change in the basis. Okay. The var scan filter option is there. You'll also get a filter file for all those filtered SNPs. And read counts, if at all, for every SNP that is occurring in that particular sequence. What is that? You'll be able to get. Finally, you need a binary calling format, BCF file. Needs to be done. So you take the samples mpile up file, you take your reference genome file, and you take the BCF tools view option, and you get this. Okay, and then this is an optional one. This is not needed. Only if you need a binary quality BAQ, then you can do this. Okay, and BCF tools view, this view file, raw file, can be converted to a BCF file. This is as simple as that, but it needs a greater precision. This is a benchmark pipeline, and this particular pipeline, I think this is our pipeline. Okay. This is the highly downloaded pipeline, and I think it has got many users. So we have benchmarked. This is a wonderful pipeline which we have used it across the tool. So you may want to refer it, by logging in. So this is the same pipeline, which you can refer, the same thing. Okay. This is how your final variant calling format is. Now this file should be mapped to different proprietary databases, which probably I will be able to show you in the evening or maybe tomorrow whenever we get time. But all these things you should probably do the exercises. Then only we can move to the next steps. It's going to be a bit rigorous. It's not going to be an easy thing. Okay. So I really wanted all of you to do exercises. So can I probably split you all so that you can also form a team and do the same kind of exercises, please, all of you? maybe if I can take the plant team into one group. Samira was there. I think Yasin is supposed to be there. I think she's not there. Yes, I think she's there. So any plant researchers around? Lipton, I think, and Esther, you both are plant researchers, if I understand right. So maybe you can join in a small group with Yasin and form and do the exercises. Can everyone please type your this thing so that maybe Raj and others will also take you to the exercises, please. Yeah. So unless you do exercises, organizing these workshops, doesn't make any sense. So whatever is your discipline, be it microbiology or the human genomics or the plant genomics or whatever, I think it's an advisable thing to do the exercises. 2 minutes before I introduce the next speaker, Raj. Yeah, please. Yeah, hello. Can I get to hear your feedback, please? Yeah. So if there are no exercises that you wanted to do, maybe I can probably move to the next part. Please allow me to introduce Tedraj Singh, my good friend. He's an associate professor and head of media affairs at JP University of Information Technology, Vaknagat in Solan, in Himachal Pradesh. His PhD from Manit in Bhopal. And then he went on to gain several years of postdoctoral years of experience in Israel, in Tel Aviv University. He raised several collaborations and he has developed several databases and tools, more precisely towards bioinformatics and evolutionary genomics. He is also a core member of Bioclues along with me, and he's also the founder of Bioinfo India, which is a portal that he has floated it, and he has put all those particular wonderful databases and web servers that he has started. And he has got an immense experience in taking up the microarray expression data, taking the microRNA and the other non-coding RNAs, which we have wonderfully exploited over the last 12-13 years. So today, Tedraj would really take us to some of the statistical interpretation of taking this evolutionary genomics upfront. We would also look into the clustering algorithms that he has developed towards this. So thank you, Raj. The floor is yours. Thank you so much.
Okay, Prashant, thank you very much. Am I audible?
Yeah, yes, Raj. Yeah.
So I think you can stop presenting so I can start my PPT also.
Yeah.
So I hope we are doing good.
Yeah, all well, Raj.
I was in the morning and I have seen some good talk from your side and it was really going good, great. And I had a class actually here at like 11 to 12, so I was with my own students. So, hello participants, good afternoon to each and everyone present over here. So I know this particular workshop is going on specifically on the systems genomic, and it's really good to see so many talks, so many things related to computational genomics, systems genomics. So today the task that had been assigned to me was on like specifically the differential expression of genes with respect to microarray data. So even I myself kept my title like this. I just want to share my screen with you so you can have a look at my slides. So my screen is visible to each and everyone.
Yes, Raj. Yeah, thank you. Yeah.
Okay, thank you, Prashant. So you can see on my very first slide lots of logos I kept here. This left, which is for my own university that I am representing, JP University of Information Technology from Himachal Pradesh, the beautiful state of India. And I will show you one or two beautiful pictures, view of our university also. Then this is the Center of Excellence in Healthcare Technology and Informatics, for which I am the coordinator for this Center of Excellence. Then I have this, my own, very own Bioclues organization, for which we are like doing from last almost 15 years. And this logo belongs to the INVIX 2020, under which we are organizing all these workshops and all these talks. So dear participants, I told you I am representing JP University, and I will show you some views also. But as you know, this particular topic that has been given to me, so I put just general like microarray data analysis. So instead of like directly going on to the data analysis, I thought let's make some background, and then we will switch over to, as Prasant told, and he gave me the responsibility to discuss lots of statistics also. So that also I am going to deal with. So as we know that this workshop is specifically on systems genomics, and we know that huge amount of data is being generated with an unprecedented rate in last 2-3 decades. And this is one of the reasons we are having to deal with this particular data for its collection, storage, as well as analysis. And finally, its interpretation towards generating the biologically meaningful information. And that is the goal of this whole exercise. And we know that our genome is encoding an enormous amount of information about our beings that can be related to our looks, our size, how our bodies work, or it can also be related to our health, even our behavior, and how we are evolving during the course of time, and actually who we are. So all these things are associated with this genomic information. So how we look, how we behave, everything is associated with this data. But if you look at this bottom part, this is just a combination of these 4 characters. So looks simple, but actually it's very complicated. So annotation of this, that's where the bioinformatics came. How we are going to annotate this data, how we are going to analyze this data. And that is where the concept of genomics came, and later on with the inclusion of systems biology and genomics, we are calling like it systems genomics. So I know my focus is different, but just to get a little background with respect to the workshop theme. When we talk about system genomics, as technologies improve, we are able to extract more and more information encoded in the genome. And that can start from very basic level, like it can be related to genes or proteins, the complexes, at the structural level, at sequence level. Similarly, we can go up to pathway level, then we can go for the network level, then the whole cell level, then the organ, then the whole community, the whole ecosystem. So there is no limit. And accordingly, if you see this middle arrow, biocomplexity is also increasing. So either it's DNA, RNA, protein, network data, it's always associated with some purpose, and the complexity is also increasing. And that's where this genomics is playing the role. Today, as we are going to discuss about the gene expression data, so I took purposely this whole microarray data analysis exercises and how we are going to do that systematically. That's the main purpose. This I am telling you about the future, because if you go to the supermarket, you pick a basket and you just go for the shopping. And this is, I am telling you, the future. You look for the organism's genome. This is the sequencing department where you have the sales. Like you will get this genome for this much amount, this much dollar, something like that. So the sale in future is possible. You go to the supermarket, you buy the genomes. Maybe the storage media can be different. So accordingly, we have to follow the procedures. But yes, it can be the future. We can do the shopping like this in future. If we are not going to see it, maybe our children or grandchildren are going to see that. Now coming to the main topic of this particular event, that is DNA microarray. So first let's have a little background, because some of you might not be working in this area. So I thought let's start with some basic, not exactly I'm covering each and every detail, but I will try to cover as much as possible. So you will feel that, okay, if you are starting from the scratch and you're going up to which level. So I will try to take you to the journey from very beginning to up to a heavy extent where you can do lots of bioinformatic analysis also. So we know that DNA microarrays, they are the devices that measure the expression of many thousands of genes in parallel. Because earlier people were working on single gene, single protein, but with the advent of technologies, we were able to identify that, okay, I can work on 5,000, 10,000, or maybe 20,000, or maybe 25,000 genes in parallel in just one experiment. And that's where the revolutionary events take place. In the past almost 3 decades, their use has grown rapidly in academia, medicine, pharmaceutical, and other kind of research related to biotechnology, agrochemical, and food industry as well. So definitely they are playing an important role. Several industries were established just to make the microarray chips. And this is how things were going on. So one of the principal feature of microarray, volume of quantitative data that they generate. Because this quantitative data is really important, because when we do analysis, normally we look for two kind of analysis. One is qualitative and another one is quantitative. Definitely both are associated with the features. Qualitative, okay, we can have features but they are not having quantity, so their accuracy can be compromised. But here when we talk about the quantitative data, and even that data is having lots of or huge amount also, then it's really good. So major challenge that was to handle and interpret this data. And this is where the bioinformatics and IT and statistics, mathematics, all computational means came. And similarly, another field arose at that time, and that is the bioinformatics of microarrays, which is like a separate domain. Many industries, many people are still working in this domain. They are doing all these experiments. I know with the advent of new technologies, it's a little bit older, but still many people, many industries are still working on, because finally the data is associated with gene expression, and that we are looking for majorly for identifying various disease processes or the entities or biomarker associated with the diseases. So just a little basic, like we know DNA micro, it's a multiplex technology, consists of an arrayed series of thousands of microscopic spots like DNA oligonucleotides, and each spot contains in picomoles a specific DNA sequence called the probes. And we can have like more than 20,000 genes simultaneously in one go. Principle is to analyze gene or protein expression through large-scale, non-radioactive northern RNA hybridization analysis. So basically, this is what people do. So this particular diagrammatic representation shows you like you have a cell, inside that we have the DNA, that is we are calling like genomic DNA. Now for this genomic DNA, we can have the mapping. Each spot contains oligonucleotide sequence of genomic DNA that uniquely represents a gene. So each spot is representing a gene. One group we are calling as the subarray or pan group, because the devices that are used to make the microarray, they are spotting them onto the glass substrate. So in one go, they are being dipped into the wells and then onto the glass, into the wells on the glass. So in one spot, we can map the one gene, but one group is the subarray or pan group. So we can have several such pan groups in an array. Now, when we talk about the disease condition or control condition, like suppose condition A, that is the test condition, or you can say disease condition. Condition B, that is the normal condition. We have to extract mRNA. Then we have the cDNA labeling with the dyes. For that, we have to put the hybridization. And hybridization is basically excitation with the laser. So for a particular wavelength, we can get the color. And final image stored as a file. So this is the overall procedure. I'm telling you briefly because this is how all the experiments are being performed. So for the disease condition, for the normal condition, because that's how we can identify between two conditions. That also we will be knowing soon. So these are the dyes that CY3 and CY5 normally used in the procedure, like you have sample from tissue 1, tissue 2, RNA extraction, mRNA, and so on. So finally, as a bioinformatician, we are getting this image, or maybe sometimes data directly from that image, not even the image. We will be provided the data directly. But we should know the background also, because when experiments are being performed, how they are being done and how the data is being accumulated. So this is done through the process of hybridization. Simply the probes on the array, they have hybridization, they have the heteroduplexes, and they are the labeled targets in the solution. So based upon that, we can identify the color, that excitation with the laser. So in this hybridization step, the spots in the hybridized microarray is excited by laser and scanned at suitable wavelength to detect the red and green lines. And according and how this color is coming, this is with respect to the wavelength, but the amount of fluorescence emitted upon excitation correspond to the amount of bound nucleic acid, and this amount tells you actually the expression level. So this is how this hybridization process is being done. So this kind of spots we can easily get, and here we can see several colors also: red, green, yellow, even black or gray sometime. So, but when we are getting this data, and there are many challenges also. Suppose this is your microarray data. Suppose there is a scratch that can also be read by the software. There can be a dust particle. There can be the bleeding of one spot into the other. So then it's difficult to identify the data from which spot is coming. But we have so many clear spots also that can be easily identified. But even when we are getting to the spots, we have this background signal. This is like pixel. We have to identify some methods to catch the exact amount of information from this data. So this spot part, this part is important. Okay. Here, if we are having this oval or circular shape, we are missing some actual data also here, and spot area to be surveyed properly. Sometimes we are getting the data, you see this part, or maybe here it's a bottom, we are getting this background signal also, and actually this is a noise. It's not a signal, it's a noise for our actual data that we are looking for. So there are many challenges for which several companies provide their own software to deal with this kind of data, or even you can write image processing algorithms for doing that. So there are many methods available for dealing with this kind of data. So these are some of the challenges when we are having the data, how we are getting it. Okay. Now, when we are having the colors, how we are going to annotate, because I know I am doing an experiment where I am dealing with two conditions. One is the disease condition, one is the normal condition. So if cDNA from condition A for a particular gene was in greater abundance than that from condition B, so one would find the spot red. If it was other way, means if opposite of that, spot would be green. So I can clearly indicate that I can compare condition A with B with respect to the spots. It is possible that gene was expressed to the same extent in both conditions. I will get the yellow spot. If the gene was not expressed in both conditions, the spot would be black. So this is how, based upon the spots that are given on my image, I can easily identify that which kind of spots I am looking for. So what is seen at the end of the experimental stage is an image of the microarray where each spot correspond to a gene has associated fluorescent value representing the relative expression level of that gene. And that data can be quantified, and that quantitative data can be useful for further analysis. Now, suppose I am working on a particular gene. Now there are so many genes with splice variants also, and they can make an impact on the array design also. So suppose a gene has splice variant, if the organism can make different transcripts of the gene by using different exons. So those combinations can give me different exon combinations, and then my transcript as well as my protein product can also be different. So those splice variants can also be a problem for designing and using microarray data. Just I will take one example of this splice variant, it will be more clear. Like I have taken this as an example. This cannabinoid receptor 1 gene. It has 3 splice variants in human. Okay, we have splice variant 1 with accession number this, it has 1755 bases. We have splice variant 2 with accession number this, 5472 bases, and we have another variant 3 with almost 1252 bases. So these three variants and how they are. Suppose I want to work on a gene and I worked on this particular splice variant, then I am missing so many information for the splice variant 2. However, some basic information will be common for all the three. And how it can be, as I told you, it's just the exons length, exons combination that we have to look for. So these are the splice variants of CNR1. Here you see variant 1, 5' to 3', and here we have like this 85 bases, 63. So here this one is missing. For this much number of bases are here in the variant 3, but some of these are missing. This is common in all three. This 1149 bases. Okay, these 120 bases are common in 1 and 2, but not in 3. So this is actually the complexity for designing even the experiment at any level. Even if you are designing primers for that, for which variant you are looking for which variant you are designing the. So you are getting some information, you are losing some information. So we have to be careful with splice variants, and they definitely make an impact. So these complications are always there when we are dealing with the genomic data. And there are many such complications, like this is one example. When when we were dealing with the human genome project, there were several goals. And one such goal was ELSI, that is ethical, legal, and social implications that may arise from human genome project. And if you look at this cartoon, person and the boss. So this is basically the employee and the boss. When the genetic profile of the employee is with the boss, then there can be problem. So boss is saying, you are lazy, you are unprotective, and frankly, you have got a lousy genome. So this can be the problem with genetic profile or genomic data also. This is just on the lighter note, but yes, definitely there are serious complications, specifically these ethical, legal, and social implications. And you can understand that when we are doing the experiments, we are always having the ethical clearance from the local level as well as other bodies. Now, once we got that data, now next step is how we are going to process the image, because we got the image, but we need to follow some steps to process the image. Okay, for that, first thing is feature extraction. Either you do machine learning, either it's deep learning like ANN, CNN, or you are simply applying SVM or other thing, random forest. We have to identify feature from the biological sequence. Similarly, for image also we have to extract the feature. Identify the positions of the feature on the microarray. For each feature, we have to identify the pixels on the image that I told you. For each feature, identify nearby pixels that will be used for background calculation, because the background signal has to be removed. That is like a noise. We have to calculate the numerical information for the intensity of the feature. So these features have to be extracted. For that, some specific computations are there. Like we have to identify the signal mean, the mean of the pixels comprising the feature. Then we have the background mean. So we can subtract one from the other and we can get the actual amount of data that we are looking for. Then we have the signal median, similarly background median. Then signal standard deviation, background standard deviations. All these statistical calculations can be done if data is being provided. Then the diameter, the number of pixels across the width of the feature, the number of pixels over there, and the flag. So these are some of the calculations like for numerical information while processing the image. Affymetrix, Agilent Technologies, and other companies, they normally provide their own software to deal with such kind of things. And definitely, no software in the world is like 100% perfect when dealing with the image data or any other kind of data. There are chances of error, but still they are working, or their basis is good algorithms, so they are working fine. So there are few steps that we need to follow systematically when we are doing the image data processing and analysis. So let's look at these systematically. So when we do image data processing and analysis, the first step in the analysis of microdata is the process the image. So I told you most scanners, they provide their own software, and relative expression level for each gene can be stored as an image, which is important to understand how data is actually being extracted from those images. So you can write your own programs, you can use the existing programs, or whatever is available to you as per your applicability. Now, the very first step is identification of the spots and distinguishing them from spurious signals. So first you have to identify the spots, and we have to remove the unwanted data. Spots are arranged like in orderly manner, maybe in sub-array or pen groups as I told you. So you should understand the pen group. The one spot is like one gene that can be generated through the TIFF image, that is tagged image file format. Most image processing software require user to specify approximately where each sub-array lies. And this information is then used to identify regions that correspond to spots. There are some methods. So how we are going to determine the spot area and local region to estimate the background hybridization. So spot signal we have to identify, and we have to estimate the background intensity also. There are two methods majorly for spot signal. The first method is where we are using an area of a fixed size that is centered on the center of mass of the spot. Computationally it is less expensive because we are focusing on the center of the mass, but it is more error prone because sometimes just focusing on the center part, the outer part we are missing a little bit, and background intensity can also be creating problems sometimes. Then we have another method that is computationally intensive and time consuming also, but that is more accurate, which precisely define the boundary of our spot and only include pixels within the boundary. It can give a better estimate of the spot intensity. Computationally intensive, time consuming. You need more CPU power, more RAM for running such kind of method. Then comes the summary state. I told you in the beginning, like there are some parameters that has to be calculated, like the mean, median, standard deviation for the signal as well as background. So how we are going to calculate the summary statistics and assigning the spot intensity after subtracting for background intensity. So mean, median, and total value for intensity, considering all the pixels in the defined area that we are selecting, because area is too much, but what we are selecting. Most approaches use spot median value that is subtracted from background median value. This is how we are identifying. So median value, normally they are working on the median intensity for the spot as well as the background. So we are looking for the pixels in the designated area, because we have to remove the background like a noise and identify all the important information for a particular spot. Then other method is to use total intensity value, which has an advantage of being insensitive to misidentification of spots, but has a disadvantage of being prone to be skewed by a few pixels with extreme intensity values. Because when we are on the center part, the intensity of pixel is high. On the outer part, it can be little low. So when you are looking for the advantage and disadvantage, each method had its own advantage as well as disadvantage. So a smaller pixel size, larger pixel size, all these things have to be taken carefully while looking for the background and the spot intensity to identify the statistical parameters. Once we identify all these values, we have to look for the expression ratio, which is the primary comparison. A relative expression level for gene can be measured by the red and green light emitting amount, that we are calling it expression ratio. We are denoting like TK, okay, for a particular gene K on any array, which is equal to RK divided by GK. RK represents the spot intensity metric for the test sample, and GK represents the spot intensity metric for the reference sample. So we have the test, we have the reference, and how we are getting the expression ratio. So for each gene, we can get the expression ratio like TK. Now, once we got the spot intensity and everything, we can get this, and based upon this primary comparison, we can identify the T median, which is R spot median minus R background median divided by G spot median minus G background median. So based upon this information, we can get the T median. Now, once we get this data, problem is transformation, because we are transforming the information, and there are chances that we are losing the information while transforming. So there are actually two types of transformations. One is lossy, one is lossless. Even lossless is not 100% lossless. Some loss is there, but little less than the lossy transformation. Lossy transformation are little easy, so there lots of information can be lost, but finally you will get some result. But lossless means there is less information being lost, but finally you will get the good results. So transformation of the expression ratio, so we can compare, because what we are doing actually here, we are comparing the higher expression as well as the lower expression. Like I want to identify in my experiment which are the highly expressed genes, which are the lowly expressed genes, and for that I want to do some computations. So that comparison, that expression ratio, the transformation, everything has to be managed very well. So the expression ratio is a relevant way of representing expression differences, and this representation can be unhelpful when one has to represent upregulation and downregulation, maybe in different ways, because we have to take them on same scale. So if we are doing scaling, means transformation with scaling is a task, tedious task that has to be done. For that,
Sir, sorry to interrupt.
yeah,
Uh, I think there's some background noise. The participants are saying that in the chat from your end. I checked all the microphones, but none of their microphones are on. I think it's coming from your end only.
okay.
If you can resolve this, please.
Actually, yeah, I am, but I am sitting in my cabin and there are some people roaming around outside. So,
Oh, okay, sir. Okay, sir. Definitely. Sorry. I
yeah,
mean, if nothing can be done, we can continue.
okay, yeah. So, thank you, Ayam. I hope it will be less now.
Yes, sir. Thank you very much.
Okay. Yeah. So suppose we are having here one example, like a gene that is upregulated by a factor of 4 has an expression ratio of 4. Okay. That we are calculating R by G, that is 4G by G. So finally it is coming 4. But for the same case, if gene is downregulated by a factor of 4, the expression ratio becomes 0.25. Like R by G is equal to R divided by 4R, that is 1 by 4, means 1 by 4 is equal to 0.25. So here it is coming 4, here it is coming 0.25. So it's really difficult to manage with the scaling of these two expression ratios for upregulation and the downregulation. For that we have some methods. That's why like upregulation is blown up and we are mapping it between 1 and infinity. Similarly, downregulation is compressed and we are mapping it between 0 and 1. So one scale we are doing like 0 and 1. For another we are doing 1 and infinity, which is not feasible actually. So this inconsistency can be eliminated by the mapping interval that can be performed with other kind of transformations. So for that we have majorly two systematic and standard transformations. One is the reciprocal or inverse transformation, and the other one is the logarithmic transformation. So those two type of transformations can be applied to take the things onto the regulated level. So if I say, okay, my upregulation is coming +1 and minus downregulation is coming -1, or upregulation is coming like +2, downregulation is coming -2, then I can say, okay, it is on the same scale. But if I say, okay, upregulation is 4, down is 0.25 or 0.23, it's really difficult to manage. So for that, we can apply the inverse or reciprocal transformation first. This inverse or reciprocal transformation, it will convert the expression ratio into fold change, which is actually the standard method that we are doing and we are using in microarray experiments also. So if the genes with an expression ratio of less than 1, the reciprocal of the expression ratio is multiplied by -1. If the expression ratio is more than or equal to 1, then the fold change is equal to the expression ratio. So this is how we are trying to manage to convert our expression ratio into fold chain. So either it's upregulation or downregulation, we can take them on the similar mapping intervals. Like here, this fold change we are trying, like tk, if tk is more than or equal to 1 or tk is less than or equal to 1. So here we are saying, okay, tk is okay, but this minus 1 by tk. So here we will get the like tk is 4, tk is 0.25, which is the problem that we were having. So by applying this inverse transformation sometime may not solve the problem. Still the mapping space is discontinuous between -1 and +1. So we can try the other one, that is the logarithmic. So it's a better transformation procedure. Here what we are doing, we are getting the log base 2 value of expression ratio, and it has the major advantage that it treats differential upregulation and downregulation equally, and also has a continuous mapping space. So if the expression ratio is 1, so log 2 1 equals to 0, which represents no change in the expression. If expression ratio is 4, then log 2 4 will be +2. If expression ratio is like 1 by 4, then log 2 1 by 4 will be equal to -2. So here we are getting what we were targeting actually. So +2, like for +4, we are getting +2. For 1 by 4, we are getting -2. So at least the final outcome, like the fold change values for these two are on the same scale, or we can say continuous mapping scale, that we can see +2 and -2. So this is how we can apply the transformations based upon the reciprocal or logarithmic. Then comes the very popular procedure, that is the data normalization, because we know that there are experimental errors also, there are handling errors, and there are many other kind of errors that are available in the microarray experiment that you are performing. So we have to eliminate the appropriate variations to compare the data, and that has to be done uniformly for upregulated data as well as downregulated data. Then only the purpose can be solved. So we have to eliminate these kind of variations by doing the normalization procedures. So the very first step in normalization procedure is, first we have to choose the gene set which consists of genes for which expression level should not change under the condition studied. So expression ratio for all genes in the gene expression set is expected to be 1 when it's not changing. When it's changing, we have to make it normalize. So we know that it's a very general term for a collection of methods that are directed at resolving the systematic errors and bias introduced by the microarray experimental platform. And if you remember, there is a standard term that is used, MIAME, minimum information about a microarray experiments. So when we are doing the experiment, those standards have to be followed. What information is required, that all is required by dealing with the data. And once we get all these first 5 steps, the 6th step, that data normalization has to be applied. So there are not many methods. I'm not covering that in detail. I will just give you some examples, like what kind of data can be and how we are going to normalize it. Suppose this is related to this C by 3 row intensity. This is related to C by 5. So we know that data is clustered over here. Now I want to take this data on the same scale, maybe onto this diagonal, but in a normalized way. So this is like before normalization, this can be after normalization. And there are several procedures available for normalizing the data. Similarly, this data for this particular die and for CI5, we know the data is coming in this format. You can write in like in descending order, or you can write in ascending order also, but still it will be biased. But we want to have it normalized. So after normalization, data can be in this form for C by 3 or C by 5 with the log base 2 intensity. So various kind of transformations, then normalization can provide you the exact data that we are looking for. It can be related to this kind of data where it is going towards like inclination and you want to make it in a linear form. Data is almost same. The only thing is little pattern will change. Here is another data. This we have used in our own publication in 2015 in PLOS ONE. So this first figure represents data before normalization, and this is after normalization. So you can easily differentiate between the two data that is available to you. And these are, the CEL files. that I will tell you also in detail. FE matrix, CEL files, for one file is representing like the one sample. So the all CEL files and same data, but normalized. So this was used in our own publication. So this is the procedure of like handling the image data, how we are getting it, transformation, normalization, and all that thing. So this is the one procedure that we have. Now comes the another important part, that is how we are going to analyze the differentially expressed gene, and we know DEG, very popular term. So differentially expressed genes. For that, we have to look for the data analysis is seen at the largest and possibly the most important area of microarray bioinformatics. But 3 points we need to discuss. Like what we are looking for here. Which genes are differentially expressed in one set of samples relative to another? If we can get answer for all these queries, means our problem is solved. What are the relationships between the genes or samples being measured? And next is it possible to classify samples based on gene expression measurement? So these things have to be carefully observed while doing the experiments, getting the data and analyzing it. We have several statistical methods, like we have to have, now experimental thing is gone. Everything we have in the form of data. Image converted into quantitative data, and that data can be analyzed by multiple means. So we are going to discuss few of them. It can be like statistical inference. We have hypothesis test, p-value, independence of data. These are some of the parameters that can be utilized while dealing with the data analysis. So statistical inference, as we all know, we are looking for a particular population based upon a sample. So randomly we pick the sample from the population, and based upon that sample we are making inference, and that can be applied to the whole population, and that is the statistical inference. Similarly, when we talk about the hypothesis testing, like it can be the null hypothesis or it can be the alternate hypothesis. We know that p-value is the standard that we need to look for, because statisticians determine whether or not a gene is differentially expressed via methodology known as hypothesis test. So basically we are trying to make a probabilistic model and want to check the hypothesis if there is any biological effect or not. So we have the null hypothesis, we have the alternate, and we have to check with the statistical means. Then another important parameter is independence of the data. Like if we are doing two measurements, so two measurements are independent if knowing the value of one measurement does not give information about the value of the other. So one information from the first measurement, second information from the second measurement, and if they are independent, means knowing first you don't know about the other, then we can say, okay, that is the independence of the data. Sorry. So we have to apply all these statistical parameters, identify some correlation measures, like we have standard correlation coefficients. If we can identify that, then we can correlate between the two experiments. So when we talk about this, we have to monitor the expression level of the genes. Patterns could be derived from analyzing this change in the expression of the gene level, and the data can be represented in the form of matrix, and we call that a gene expression matrix. So this data that is available to us can be in the form of expression levels. Now, when we represent this data in matrix form, you know, matrix, if I say, okay, 2D matrix, it's simple row and column combination. It can be like m by n matrix. So each row in this matrix corresponds to a particular gene, and each column could either correspond to an experimental condition or a specific time point at which expression of the gene has been measured. So row and column format for a particular gene and for a particular experiment. This kind of data we can have. Expression level for gene X for different experimental conditions are cumulatively called the gene expression profile. For sample, we can have the sample expression profile or experimental conditions. So either gene expression profile, sample expression profile, we will have the matrix. row for genes, column for the experiment. So we will represent data in the form of gene matrix. Like here, this first table tell you the absolute measurement. This like row means first row, second row, third row, fourth row. So gene A. Column 1, column 2, column 3, column 4. That is the condition, means experimental conditions. So for condition 1, data for all the genes. For condition 2, data for all the genes, and so on. So this is the absolute measurement for a particular condition, for a particular gene. This is the relative measurement. Like here you have gene A, B, C, D, but here you are trying to identify C1 with respect to C4, C2 with respect to C4, C3 with respect to C4, and so on. So we can identify the absolute measurement as well as a relative measurement for all the conditions in the experiment. I hope you are following me. So then we can have the log2 relative measurement and the discrete values for the same. So we can calculate the log2 value for C1 by C4, C2 by C4, C3 by C4, and similarly the discrete values can also be calculated. Because if we have the expression ratio with the help of log2, then if the value is greater than 1, change to 1. Genes with value less than -1 were changed to -1. So here discrete value normally we are applying like in the 0, 1 or -1 format. So +1, -1 and 0. So any value between -1 and +1 was changed to 0. So all these three conditions can give you the discrete value. This is the absolute measurement, then you have the relative measurement, then you identify the log2 value for that relative measurement, and then discrete value from this table, not from here. Okay, remember, so 1D is coming from 1C. 1C was coming based upon the 1B, and so on. So once we get all these values, we are representing like the expression profile through vectors. So vector, we know that we are having n-dimensional space, and here we are talking about the m by n matrix. And vector, we know that vector is something that can give you the direction as well as the method to deal with that data. So then your data is directional as well as having a method. It's not something that is just kept like in a black box. So vector is having some specific purpose. So gene can be considered as a vector in n-dimensional space. We are looking for this m by n matrix. So we can represent like this. This is the matrix x11, 12, 1n. Here we have 11, 21, 31, and m1. So in this way, m by n matrix can be filled. So suppose you are looking for the expression profile of particular gene I, you can represent as a row vector because genes in row. So gi is your xi1, xi2, xi3, and so on up to xin. Similarly, if you want to have expression profile of a sample J, you can represent it as a column vector. So basically, this matrix you are representing in the row vector as well as column vector. So here you have x1j, x2j, x3j, and so on. Okay. Now, once we represent this in the vector form, like we have the measurement in the form of matrix, so now we can perform some computations over it. So we can finally get the results. But expression analysis can be done by using some distance measures also. There are so many distances that are being applied even in bioinformatics. If you look at, so many distance measures are being there. If you look at this edit distance, that is like Euclidean distance, then we have the Hamming distance. And those were also very much implemented in alignment algorithms, either you are applying through dynamic programming or other means. So distance measure can be applied for comparing the gene expression profiles or sample expression profiles because they are the standard measures, they are available to each and everyone, and they are already proven, so we can usually utilize them. So variety of distance measures can be used to calculate the similarity in the expression profiles. Like here I am just giving one example of Euclidean distance, which is actually the square root of the sum of the square of the distance between the values in each condition. So Suppose we are having this Euclidean distance for B we have B1 and B2. So you can calculate the Euclidean distance between A and B like the square root of sum of A1 minus B1 square plus A2 minus B2 square for both the conditions. So we know that Euclidean distance is standard measure. Same can be applied on R sample data also. This is just a representation like on when you are looking for the space, how it will look like. So Euclidean distance between x and y, and this is the distance you are looking for. But this can be calculated using this formula only. If you have multiple points in the space, then you can go with this. Like x1, y1, x2, y2, x3, y3, x and y. So then comes the another standard measure. Okay, if somebody asks you, okay, when you are correlating the sample, how you are measuring it. So okay, you can say, okay, I will give you the correlation coefficient. So it can give you, okay, if it's coming like 0.7, 0.8, 0.9, or maybe 1, how the samples are correlated. If it's coming like 5, 0.5 or 0.4, means they are not so much correlated. So this correlation can be established again with the standard measures. So we have this Pearson correlation coefficient that can also be measured easily. So this is one of the most commonly used metric. So if you have given the expression ratio for 2 genes, suppose, okay, under 3 different conditions. So these are the 2 genes A and B. Your conditions are 1, 2, 3. So A1, A2, A3. For B, you have 3 conditions, B1, B2, and B3. Then how you can calculate PCC? Simple. First step, compute the mean. So for the A bar, you will have A1 by A2, A3 divided by 3. Similarly for B. Then the mean center for both the conditions. Next step, AOB divided by modulus of A bar into B bar. This can be calculated by this formula, this by this, AOB by this, because A bar and B bar we have already calculated in the last step. So based upon that, for each I is 1 to n, for n number of things, you can easily calculate this. Then you will get the PCC value. If it is coming 1, it means the 2 genes are exactly similar expression profiles having. -1 number means the 2 genes have exactly opposite expression profiles. And 0 means no relationship can be inferred between the expression profile of the genes. Normally, in reality, if you look at PCC, that will range from -1 to +1. So if suppose you say, okay, PCC value more than or equal to 0.7, that genes behave similarly. And PCC value less than or equal to -0.7, it suggests that genes have opposite behavior. So based upon the PCC, we can correlate them. This is how we go systematically. Okay, suppose now I have some quantitative data with you. So consider 2 genes with an expression profile. Gene A with 13569 and gene B with 26912 and 90. Now you want to calculate PCC between these two to identify if their expression profile is matching or not. We know the values are different, but we just need to check it. So what we can do? We can do same thing. We identify first A bar, B bar for both. Like this plus this plus this divided by, we have conditions are 5 here, remember. So condition A1, A2, A3, A4, A5 for B1, B1, B2, B3 and B4 and B5. So we will identify these parameters A bar, B bar, capital A and B bar, then AOB, your A bar modulus, B bar modulus, and based upon that you finally identify the PCC, and that is coming 0.995. So I'm sure it is significant. So this is coming good correlation between the profiles. So means good profile means they are behaving similarly. So this is how all the statistical computations could be applied on any gene expression data that is being provided to you. And we have started from like microarray experiment data coming to identification of correlation coefficients. This journey starting from experimental data, getting the image, processing that image, getting the quantification data, after that coming on to that. And try to compile each and everything. It was a little faster because in 1 hour or so I have to cover so many things. But I'm sure this will give you like how things can be done. There are many tools available for analysis also. Now, when we talk about like dimensionality reduction, PCA comes into the mind on a very first note. Here, this example is taken like this. When you see this particular thing, you can see it can be like a picture, it can be something else when you are seeing the image. But when you see this second image, it can be like, okay, it can be even a candy stick, thermometer, or anything. But when somebody is on third image, you can think, okay, it looks like a pen. So viewing same thing from three different angles provide you the clarity when more components are added to it. This was having the first component, you can have the second component, you can have the third component. Means you want to see the things in multi-dimensional space. And when we say PCA, principal component analysis, is we are trying to reduce the dimensions of the entity that we are dealing with, and we are trying to focus on important components, principal components, which are actually representing the more percentage of our entity. This is how PCA can be implemented on this kind of data. So once you have identified all this data, like this, like first component, your second component, it can be in multiple dimensional space that you want to deal with. Suppose you have the standard deviation, proportion of variance, and cumulative proportion for first component, second, third, fourth, and fifth. So this is how your first component is representing, second is representing. So you see this 77% cumulative proportion represented by first component, then the second, then the third, fourth, and fifth. So in 5 components you are representing the 100%. But if you look at this carefully, 77% by first, second 11. So 2 are making almost 88%, and remaining 3 are making 12. So if we can cover first and 2 component, 88% of our entity can be covered. We can ignore these 3. So instead of 5, we can deal with the 2 components. This is how we deal with the principal components to look onto a space. एक experiment है, I will give you this first. It was performed on the 1000 most varying genes from a data set in order to be able to visualize the similarity among the 6 samples. So 6 samples with multiple R's, 0.5 R, 2 R, 5 R, 7, 9, 11. Represent that. First thing is like, okay, this is the first principal component, this is second. So based upon this correlation, I have to correlate कि मेरा जो experiment है, अगर मैं इन दोनों को close करता हूँ आपस में, then I am missing lots of information. If I cover these 2, again I am missing. So I try to compile the data in such a way so I can collect information. So I can have like, okay, these 3 experiments are correlated to each other, and they are representing most of the, because it's like 11-hour experiment, and from 7 to 11 the data is coming. So PCA can give you some clusters. When you want to classify your data, when you want to cluster your data, that cluster can be of significant information. यहाँ पे कोई cluster नहीं बनेगा, ये independent जैसे बनेंगे अगर आप बनाओगे भी, because when you want to cover the clusters, you cannot draw a zigzag line. It has to be in some uniform scaling format. So oval or circle are the best method to correlate these points. So this is making one spot or one cluster that is useful and can provide me some important information with respect to the first principal component and second. So PCA can be applied. Similarly, there are many classification and clustering techniques that can also be applied. I haven't included all of them because due to the time limit. This example I'm showing you from my own publication that we did in BMC Genomics like 10 and 11 years back in 2009. It was on the tunicates. So we were trying to study the evolutionary genomics of tunicates using 13 mitochondrial proteins. These are 13 protein coding genes from these tunicates. Because one of the tunicates is Hardmania momus, I sequenced the complete genome of hydropaniomonas and submitted it to NCBI. And then I collected available genomes, their information regarding mitochondrial genomes and their 13 protein coding genes, and tried to correlate them. And you can see here, the classes are coming in clusters. If this is cephalochordata, so they are coming together. Similarly, they are also making some clusters. Some are coming randomly also. This porifera and nadaria. But if you look at this carefully, all the tunicates were coming. So based upon that, we proposed a new hypothesis over there. And we have taken the 54 taxon metagenome dataset for all 13 protein mitochondrial genes. So, but PCA when implemented, this first principal component represents 53.1, second 15.3. If you look at the perfect example, it's not a very perfect example because two components are representing almost less than 70%, which is not as accurate as it should be, but still we were able to identify a particular pattern based upon PCA analysis on mitochondrial genomic data. So this is how the systematic genomics data can be implemented for various kind of mitochondrial genes or proteins or any other kind of data that is available to you. So now coming on to the third section of my talk, there are many databases and analysis tools available. I told you that Affymetrix is providing you technology. Sometimes the data is also having the written over there like AT. Your file is having the extension or like the underscore AT. So that AT is like Affymetrix technology. So we are having their own technology. Several tools are available, but Affymetrix provide you a special file that is the .cl files, which contains all the data regarding your experiments for microarray. So when you are looking for the gene expression omnibus GEO database, you just download CEL files. Means that is Affymetrix DNA microarray image data being converted into quantitative form with their own software, and that data can be utilized by multiple softwares all over the world for analyzing that data. Because you have the CEL data means you have everything related to that experiment except one thing, and that is the CDF file. So for that, we have this CDF file that is called chip definition file. So everything related to experiment should be on this chip. And these chips are sometimes experimental, condition or organism specific. Like for human, they provide normally 8 to 10 CDF files only. So out of these 8 to 10 CDF files, any one of them can fit on the CEL file. CEL file, when you are getting the experimental data or you are reading the paper related to that particular experiment, always they are writing that we have used this particular CDF file for the experimental purpose, means for collection of the data finally. So that CDF file you can easily download. That is also available on your GEO dataset. And then with the combination of CEL and CDF, you can do analysis with lots of softwares also. If you look at the specifically, just quickly, the CEL data format, file format, that will have the CEL, the header, the intensity, masks, outliers, and modified. These are some of the fields. And this format has ASCII text file, which is similar to Windows initiation file, Windows INI file. So I just put some screenshots from the CEL file. If you look at like the tag, like version, the version number always set to 3. Then the tag is like column, the number of columns in the array of the cells. Rows, the number of rows in the array. Total X, total Y, and all these things means all this information. So this is your tag and its description. So all the tags and the description can be available. If you want to read more about this, like the number of cells, cell header, and all these things, and their description is also available. So this kind of data is available in CEL file. So it's like a standard format. If you see the file, you will see only the X, Y, the mean, standard deviation, the number of pixels. Only 5-6 columns will be there. But rows kitni hongi wahan pe? You will have the rows like if there are 20,000 genes, you will have 20,000 plus genes, rows. 20,000 rows for each gene, because I told you when we are representing it in the form of matrix, rows will be the genes and columns will be the conditions for the experiments. So this is how we are getting the data. Now, if you look at, I told you many tools are available, and this is the tool that we have developed, it was developed in collaboration with one of my collaborators in Morocco. So Morocco's team and my team from GUIT, we developed this is R-based tool that is available over here. If you are knowing very well R, then you can easily download it bioinformatics.org/WBDEGS, because it was based upon a new methodology that was within between group differential expression of genes. So this tool is available on the website. I will just show you the website also. When you install it on your R system, you will see this like run app. So you click over here and you can run this software. So I will just show you. I have opened this. So this is the home page of this particular website where this methodology is given. Input your CEL file, the pre-processing will be done like the background correction and normalization, then DEG, differential expression analysis, like within between approach, statistical testing and gene filter. Then you will identify the expressed genes and then you can visualize them with the help of Venn diagram also, so you can compare with the multiple methods. This is like the installation procedure is given on this website. You can just follow that, which packages you need to install them. If you want to run, how you are going to run the WBDGS, like this, all the help is given, run in the window, within between differential, pre-processing, gene selection, these are the steps. First you upload the data, do the pre-processing and then the gene selection. Here you have the gene selection plot and the Venn diagram also. This is the hands-on that is given to you. So data upload, pre-processing, you can select some methods that are being implemented, pre-processing, and here you have the gene expression matrix, gene selection plots, Venn diagram. So you do the selection, and you see here this is a probe ID actually. So one probe ID is reflecting one gene, and _80 this represents the Affimatics technology. So you do the gene selection, and gene selection, like we have groups. Suppose you are having one experiment where you have control and disease condition. So you can have one group for the disease, one for the control, and then you want to compare them. So one CEL file representing one sample or one experimental condition. So you compare them, make the groups, and our tool will do all the analysis. You can choose the method also, like you want to do simple statistical test, twilight, linear models, or SAM, significance analysis of microvariate. So using all these methods, you can generate simply the volcano plots. If you are comparing them between the groups, like this, within the group, less deviation will be there. Then you have the cutoff for the fold chain, the p-value cutoff can be there, significance analysis of microvariate SAM plots can also be generated like this. And you know, these plots can be easily published also. You can publish them as your results. If there is some problem in running, means there is some problem in the input data. If input data is correct, that has to be checked carefully. After that, you can run all this twilight, linear models, and then you can have various kind of Venn diagrams also. So this kind of Venn diagrams can also be generated where you are comparing all these models, and you can see easily that how many genes are coming in the combination of 1, 2, 3, and 4 models. Individually for L models, only 206. For SAM and twilight, independently, nothing is coming. So your more focus is toward these two experiments or methods. So this is how you can use this easily. Availability and requirement, you can download it from here. Some references are given. So this is how, this is just one demonstration I gave you. So if you are compatible with R, you can easily download it, install it, and utilize it. Okay, coming back to my presentation. So this is done WBDGS. This is the procedure that I already explained you. Then there are many public databases available like GEO, most popular, Gene Expression Omniverse. Then we have Stanford Microvariate Database, UNC Microvariate Database. So there are many databases available that can specifically be targeted. MUSC Array Express from UBI, very popular. So these are some of the databases that are publicly available. Similarly, there are some tools. Some are open source software like Bioconductor. We are already using GeneSpring, NetFX from Affimatics. So these are some of the tools that are available. Some are free, some are with the registration procedure, and some are commercial. Some more tools are there. Even Ingenuity, Geneinvestigator, these are some payment based. Like Panther, it's free. So there also we can have. Besides that, there are many other tools which are available for microarray data analysis, or maybe sometime we want to apply ontological analysis with the gene expression data, because out of 20,000, suppose you identify 20 genes which are differentially expressed. So those 20 genes you want to get their ontology also. For that, there are some tools like WebGestalt is there, Gorilla is there, and there are many other tools that can also be utilized. So I think it's not the end, it's not even the beginning of the end, perhaps it's end of the beginning. This is the given by Winston Churchill during World War II. So just some acknowledgements. First, my own very Bioclues organization, my own department, my university, DST, DBT and ISMR, from where I got various funding. Even one ISMR is running nowadays. Then the organizing committee of NGX, specifically the Prasant, my very close friend, Google and other open sources for the data and various images, and own team. Here, these all are my PhD scholars. They all completed these, recently completed. These two are pursuing PhD, Rohit and Navin. Rest have completed PhD. This is doing postdoc in US. Ankita and Ashwini are working as RA. Monica was NPDF with Dr. Raghava at Interaction Leader. Priya is in Netherlands. Nupur has joined the Institute of Bioinformatics, Bangalore. So this is my team, and definitely now I would like to show you the beautiful view of my university also. So see here. The pictures give you of my whole campus of JYT. Thank you very much. If you have any queries, please you can write me in the chat box, or even you can drop me an email if you want to connect. Thank you very much once again.
Yeah. Thank you very much, Raj, for a wonderful presentation. It was absolutely awesome. It has really set up a kind of a precedent to look into the differential express gene analysis, taking the microarray front to the next generation sequencing that Ashwini is going to deliver now. It's absolutely very terrific. So the forum is open for a quick Q&A session, please. Despite Tedra's voice, pretty bad, he has really taken his wonderful time. Thank you so much, Raj.
Yeah, thank you, Prasant. So if even you are not having query at this time, please send me through my. You can send me on my email also. And I'm sure I tried to give you something interesting that you can utilize in future, near future.
Yeah. So if not, let's thank Tedraj for his wonderful presentation. Thank you so much, Raj.
Okay, thank you.
Yeah. Thank you, Raj. Yeah. So if you can just stop your screen, Raj.
Yeah, sure. I just stopped presenting.
So please just allow me to Ashwini Kumar Mishra, who is the director of DNA Experts. He's also another good friend of mine. He's absolutely very busy man. In fact, next time I'm just going to talk to him only in Telugu. This time, I thought I wouldn't talk. So he's also a co-PI for a project, on vitamin K, and he has got vast experience in analyzing the next generation sequencing based approaches. He worked for Agilent, and he has been instrumental in developing various assays, including the NGS based panels as well. A wonderful niche working for both wet lab and as well as dry work. And most importantly, is the director of 3 companies, 2 based in Delhi and 1 currently where he has really established working for SARS-CoV-2 diagnostics at Andhra Pradesh, says, which is a special economic zone thing where he is trying to diagnostic based kits. So without wasting time, I would request Ash to come up with his presentation on differential express gene analysis, more relevant to DSeq and more relevant to the principal component analysis and the clustering algorithms that is going to demonstrate. Thank you, Ash. Please, yeah.
Thank you, Prash. Thank you for such a wonderful introduction. And so I'm going to take up this DSeq part. So my part incidentally coincides with Ankita's part also, who is also going to talk about DSeq. So probably, I'm not going to go much in detail. Perhaps I'll leave that for Ankita to complete. So I'll, go from, go across different modules of DSeq, uh, give a hands-on demo on usage of DSeq, and then followed by questions if anybody is having. So when we talk about next generation sequencing data analysis, uh, specifically in next generation sequencing, if we talk about RNA-seq, then the first thing that comes after the raw data is QC. Now, once QC is done, then alignment, which has been already covered which is done through usage of different softwares. Now, the next step is knowing, what is the expression level of genes and how the genes are.
Yeah, sorry, Ash. I'm sorry. Are you sharing any screen, Ash? I'm sorry.
I think Karan would be sharing the screen.
Okay. Yeah. Thank you. Yeah, thank you.
So this RNA-seq data that we need to analyze to figure out what genes are expressing and what is the expression level and how they are expressing in comparison to the same set of genes in different set of samples. So there are multiple tools available for doing this comparative differential expression analysis. One of the most widely used tools for this is DSeq. It has got two versions, the DSeq and DSeq2. The DSeq essentially is the first version of the software, and then DSeq2 is the later version of the software. Basically, it's a tool for hypothesis testing and figuring out differential gene expression analysis in RNA-seq data. But since the purpose of this is to figure out differential expression analysis, it has been used in many other applications apart from RNA-seq as well. So this DSeq applies the negative binomial distribution and a likelihood ratio test. It normalizes data by trimmed mean M values, and it incorporates information of all genes in a set of samples. So essentially, to start with DSeq, we actually need the count matrix file of genes. So this count matrix file is obtained through the previous step wherein the alignment of the reads to the reference genome is carried out, or even in case of de novo, when there is de novo assembly, then this alignment is done to the de novo assembled mixed genome, and then the count data is obtained. So to start with this process, you need a count data. And then I have my colleague Karan over here, who will take up from here for all the steps in DSeq analysis. So Karan is going to take hands-on exercise from here on DSeq.
Thank you, sir. I will take from here. So first we have to do the quality control analysis of the FastQ file. So to do this, we have to use the FastQC software. This uses the thread score algorithm. what is self basically it is a probability of incorrect base score means if there is 10% quality score, so there is incorrect one basis in 10. For 20 there is incorrect one basis in 100, and that's go on. So we use a 20 threshold value. Lesser than 20 threshold value is called a bad quality. The graph which was showing in right side, uh, and a greater than 20 score is called a good quality fast files. Then after that we find that the quality is well enough, then we move forward for the alignment to the reference file. So we use this HiSet2 software, which is with- which align the reference files and produce a SAM file. So in alignment summary, there are two type of alignments. There are two types. For unpaired, the smaller one, left one is summary zone, uh, and the right one, the larger one, is for paired and summary files. So for after alignment, we want to sort the BAM file so that it can be used in further steps. So we use the align SAM file to convert into BAM file, and then sort them. After alignment, then we want to assemble and estimate the abundance to produce the count file. So this is done by string type, which is assembler for RNA-seq aligned data to a transcript. So it takes the BAM file input and produce a GTF file. Basically, GTF file is a 9-column file which contain every gene, chromosome, start and positions, and FPKM, RPKM values. Uh, so after getting the estimate the abundance, uh, we want to get the final count matrix. For this, we have a script called PREPDE, a Python script, which convert the GTF files into a matrix of counts per genes. So further, after getting the count file, we want to differentially test the R genes against the sample. So we use the DESeq tools, which works on negative binomial distribution method. Uh, Negative binomial distribution. So from here, I will demonstrate the processes. So first we will import the DESeq package, and this down to package are for the table manipulation. After importing, we want to import our count data, which we have got from the prepde.py script. And this is how our count table looks like. Here there are two groups, replicates, three unaffected groups, and three infected groups. After that, we want to clean our data, because if there is in all the columns, if there is all the zeros, this will create a uh, distribution in our analysis. So we want to remove the rows, all the rows that contain zeros. After that, we have a call data. We have to create the call data columns that contains which are with the metadata column that contains which are control, which are infected, and then differential expression testing. The first line of the script says that row names call conditions of call. This will check that row names of conditions. This conditions is equal to row names of experiment data. This data, and then we want to change the column names of the count table, and then we further check. So we once again check that the row names and column names of exp is same or not. Then we create a summary file of DEC matrix, and then we calculate the differential test analysis, and then we create a result table. The result table are shown here. In this table, the p value, PADJ value, and log2 fold chain per gene are given. And then we move forward to a MA plot. The MA plot uses the PADJ value and the base mean of the above RES result to produce this plot. This the red spot. This every point is a gene, and the red spot indicates the PADJ lesser than 0.0, means these are the significant genes, and the middle red line above this are upregulated, and lower this are downregulated genes. And then this is also volcano plot. Volcano plot tell us about how many other significant genes and how many upregulated, downregulated genes are present in our data sets. And then we want to scale our data. Logarithmic scaling of the data we carried out using rlog function of the DEC2, and then we plot PCA according to the conditions. So
yeah, can go ahead.
So in this wait a minute. So in this interpreting the PCA plot, so we produce two conditions. The PCA are normally shows the same groups, means clustering the same group, like we have in this. We are in the red spot. This red spot cluster together because of they are replicates, and the green spot are somewhat clustering together because they are replicates. So in PCA we check that our data set, how much variable our data set is. And the final point, it should be it should have the cluster of the cluster is well separated along. So this cluster, purple cluster and the orange cluster, they are variable in the PC1. So this shows that the 33% of women, this samples is much variable than this, and PC. And the PC2 shows the intra group relationship. Thank you.
So Karan, can we come into command prompt and show them all the scripts?
Means the script.
Yeah. Yeah. So Ash and Karan, can I ask one question, please? Yeah.
Yes, sir.
Yeah.
Yes, please.
When we look into the counts, I know this is, when we have a pair of samples, or probably, some samples where, we really wanted to differentiate between some, unhealthy cohort. But what if there are replicates how do we come up with a principal component, if we have, replicates across?
Raj, basically when we do principal component analysis, so like first thing is to do QC of your data using principal component analysis before I would say even looking at differential expression sheet. So principal component analysis is basically a measurement of variance of the samples. Generally, it is thought that PC is for finding out similarity between the samples, but it is exactly opposite. It's about finding out the variations between samples. This principal component analysis can be 2-dimension, 3-dimension, 4-dimension, 5-dimension. It can go endlessly. So it depends on somebody's own understanding and their requirement. Who requires how many dimensions of principal component analysis? Or I would say something as complex as next generation sequencing data, where we are looking at around 20 to 25,000 genes at a single stretch, and their pattern across uh, I would say biological replicates or technical replicates or type of samples or type of experiment. Uh, Generally, a 3D PCA is good enough to differentiate, but 3D PCA is not the end of it. So if you want to have even higher stringent kind of criteria for finding out how the samples are behaving in terms of the expression levels, so then even uh, higher dimensions could be applied to know about those expression levels.
Right. So basically, I mean, if you really look into the so-called, the fragments per kilobases per million read file, which we generate, from cufflinks or whatever. This is basically the number of counts that we are trying to use it, with that Python script that Karan was demonstrating, right? So that can also be cumulative, right? That can also go along with several replicates or several data without incurring any bias.
Yeah, the idea of having FPKM or TPM or TMM, these are various normalization algorithms. So the idea of having them in place is that since we have data sets from different samples having different amount of data. So
Right.
let's say we have one sample wherein we have data of 10 million reads, another sample where the data would be 15 million reads. The third sample would be having data of 18 million reads, because in NGS you cannot actually control that all the samples should have exactly similar number of reads. So based upon titration, which is done just before next generation sequencing starts, the amount of data generated per sample can be very similar, but they cannot be exactly similar. So before we do any sort of comparison of inter samples, we need to bring them to a same platform to compare them. So that platform is called a normalization. It can be done through, previously it was done through RPKM, now FPKM or TPM or TM. So it depends upon, one's easiness, which one, one understands and which one prefers. We really, as a commercial provider, we go by what, people demand. FPKM is the one which is most widely used. So even we use mostly FPKM, but, TPM and TM are also kind of required at couple of instances.
Right. Yeah. Perfect, Ash. Thank you. Any more questions, please, before Karan moves forward? Karan and Ash. Yeah. Shalini or Nidhi, any questions, please? Madhusmita. Yeah. So there are no questions. I think please go ahead, Karan. Thank you. Yeah, thank you, Ash.
Do we have Galaxy session today?
Yeah. Please, Raj Thakur. Yeah, please.
Do we have Galaxy hands-on session today?
Yeah. We have Galaxy. I mean, we'll also show you the RNA-seq pipeline using Galaxy. IAM will show you. Yeah.
Okay. Thank you.
Hi, is Karan still here?
Yes, sir.
Sorry, I'll just mute my other speaker. Yeah. So I was asking that whether we can. I just posted that in chat as well. Is there any way that we can do DSeq analysis without replicates?
Yes, we can do using HR packager. This takes the uh, this take without replicates. But by this package DE62 we can't. There is a limitation that it requires only replicates or duplicates.
Okay, so DSeq2 basically requires replicates.
Duplicates, yeah, replicates. But HR can do.
Uh, okay, okay.
That also
So
makes a slightly different question, Karan. I know Edger has got a kind of a limitation because at any given point of time, you cannot correlate the what do you call, the number of counts across the different
Across
subpopulation.
groups.
Yeah. So how do we overcome this? Because at any given point of time, say for the same data sets, if you run couplings, and I think say DC or Edger or BASR or whatever, the number of counts, irrespective of the statistical correlation, like Ash was saying, even if you take the FPKM across,
Yes, sir.
we are going to invite a lot of bias on this. So how do we overcome this?
It's difficult to answer. How do we overcome this? It's difficult because each software has their own way of, I would say, normalization and then calculation both. So in our experience, what we have observed is, for a set of data, if we compare DSeq versus EDGA versus Lima-Boom, uh, we have seen that DSeq generally gives much better results as compared to EDGA or Lima. Uh, Drawback of EDGA and Lima is they give extremely large number of statistically significant number of differentially expressed. That is a limitation in I would say Lima. Even one of the but
Yeah, Ash, I'm sorry, your voice is breaking, I presume. I don't know. The connection is a bit low at your end. Yeah.
that most of the things. Hello.
Yeah. Thank you, Ash. Karan, please go ahead. Thank you. Yeah, hello. Yeah, hello, Ash. Is it audible? I'm sorry. Hello.
Yeah, Prashant, for you it's audible, but I think we cannot hear Dr. Soni.
Yeah. Yeah, good, Raj. Yeah, I think Karan has also lost connection, I guess. Yeah.
No, sir, I'm here. I'm here.
Oh, Karan is here. Okay. Yeah. Sorry. Yeah.
Yeah, Karan is here. I think.
Sir has lost the connection. Ashwini, sir.
Yeah.
Okay.
So in the meantime, maybe if I can request all of you to switch on your camera, please. We'll all have a group photograph, please. Yeah. I think Ashwin is also here. Please switch on your camera for one group photograph. This is a kind of a rare opportunity where all of us are here. Yes. I'm a good photographer with all the lovely smiles I can see. Yeah. Yes. Others, please. Yeah. A quick photograph. All of you. I mean, there are still Sundarda, Rashmi, Ashok, Sonal, Kikrusenyo, Shalini, Ayam and Nidhi and Shalini, and of course Rajul is here. Himani, Mehak, Hemani Sharma. Please, yeah, one quick photograph. I'll just take one. Ashokji. Yeah. So I think I'm just going to say cheese. Yeah. Thank you. Yeah. One minute, please. 1 minute. Yes. Say cheese. Yeah. Thank you. Thank you very much. Yeah. Sorry, Ash was saying something. We got disconnected. Yeah, please. Yeah.
I was just saying that DSeq actually in comparison to others, it gives much better result as compared to either HR or Lima. In most of the cases, I would say, like almost 90% of the cases. You're on mute.
Press, you are muted.
Yeah, sorry. Yeah. So if you really look into the previous cases, Ash, for example, the GPM data sets, if you remember, which we analyzed some 3 years ago, that was the so-called pre-benchmark cufflinks pipeline, although I ran DSeq as well. And now, from the recent Jyoti's data sets as well. I know it will invite really bias, but at least the fragments per kilobasis per million reads for the same sample across different algorithms should be okay, isn't it?
Yeah.
That was my a very stupidest question, I thought. Yeah.
I did not understand the question.
No, what I said was if we get into consideration the fragments per kilobasis per million reads, FPKM values, for the same samples across different tools, that which we are going to generate from the GTF, it should be at least the same, right? For the same samples, the algorithm wouldn't change.
Yes, the formula should remain the same.
Yeah, but that is also my point. Why is that? We are not getting that precision. That was my point. Yeah.
I don't have a right answer right now. I can probably dig up and get back to you.
Yeah, I understand. Yeah. Thank you, Ash. So is Karan going to give a demo, Ash? Anything else, or any online server or something, or is it done?
Karan?
Yes, sir.
So is the demo possible online right now?
Linux demo is not possible. That's why I have not given that part.
Yeah, no, it's okay. Yeah, it's okay. So,
these you have
what
given.
what we can do, Raj, is since we have some time left, uh, we can, we would need 10 minutes to set this up on our server because, Karan is working remotely right now.
right,
So,
yeah,
and then we can do this.
yeah.
Or, next session, which is there, after that session, we can take this up.
Yeah, sure, Ash. No, wonderful. Thank you so much. And before I forget, Ash, Karan, and Tedraj, I hope you don't mind if we record this session, right?
No problem.
Yeah,
There's
yeah,
no
no problem,
proprietary.
no problem.
No problem, sir.
Sure.
No problem.
कंपनी वालों के साथ काम कर रहे हैं, especially Ash. I hope you wouldn't mind. Yeah.
No,
Anyways, yeah.
but this is, this is an academic session, no? So,
Yeah, right. No, I was just
company
joking.
people
Yeah,
are,
I know.
company
I know
people are
you.
also using Skype. They can be a part of academic sessions as well.
Right. Yeah. So it's automatically pre-recorded. I know. I read your mind, Karan's mind and Raj's mind. It's if formality के लिए मैं बोला हूँ बस. Thank you so
No, no,
much.
no, it's okay. And
Yeah.
you have recording for this consent also, that we give our
Right.
consent.
Right. Yeah. Thank you so much, Raj.
Okay.
Without wasting time, I will invite Ayam to give a demo on the cufflinks pipeline. Which probably which we didn't. I mean, he'll also probably give us a demo in the command line as well, which we have benchmarked, and he'll also demonstrate, in the Galaxy which we have worked in the morning. So Ayam is an MTech graduate, a BTech and Tech integrated graduate from Rajiv Gandhi Pradyogik University, Rajiv Gandhi Technological University from Bhopal, Madhya Pradesh. Then he did his project with me for 1 year, 3 months. He was a wonderful researcher. At the helm itself, we got two papers. One frontiers in genetics, and he has benchmarked 2 or 3 other tools as well. And then when we got this vitamin K project, we hired him. So he's going to give us a kind of a demo on both Galaxy, where he would show the RNA-seq cufflinks, and as well as the command line as well. Ayam?
Yeah, thank you, Prashant, for the wonderful introduction.
Yeah.
I think we shall start with Galaxy first, if that is okay with everybody. Show everybody how to set up the account if they haven't done that yet. It will take 2 minutes, and then we'll proceed further. Share my screen with you guys, and then we'll move on. Screen is visible, right?
Yes, I am. Go ahead. Yeah.
Yeah, okay. So you guys have to use the URL. I'm just posting it in the chat box here. So just visit usegalaxy.org. Okay. There's a user section here. Here you have to sign up. I'll just show you how to sign up on the website. You log in, you will be greeted by this awesome page. It looks very good. Then you have to click on login or register. If you are not already registered, just enter your email. Just click on register here and enter all these details. Two-step process. As soon as you click create, you will get a email confirmation through that. So just let me know in the chat box or with the microphone on so that I can move further.
I was just saying that they already have used the Galaxy account. Some of them have already done the access since morning. So maybe you can just go ahead with
Yeah,
the
I
demo.
was
Yeah.
for a confirmation. Sorry. Yeah.
Yeah.
Okay. So as soon as you guys log in, you're again greeted with this page. This is your history pane. In Galaxy will be shown on this page. I've been working on Galaxy for a while now, so the history is very cluttered. I'm sorry. This is the tools pane. Here you will get all the tools that you can use. So we'll use a few these sample data sets for which I can give you guys the links. Yeah. I'll just post the links in the chat box. These are small data sets, 111 MB. Download them from your local machine if you are working there. It will not take much time with the connection. So data sets, or maybe we can also use the shared data from the libraries, data libraries. But I am not using the shared data from the libraries because it will take process all those things. So this is a small data set that is containing only data from one chromosome. Because it was small, so it produces the results quite fast. So in this data set, there are replicates of a normal tissue and a tumor tissue. In total, there are three replicates. So when you guys download it, and the way to upload it is just get data. You click here and the get data option. Sorry. In the get data tab, you just click here, and then there's the option for uploading files from your computer. When you click here, just select choose from local file and select whatever you want to upload. Uploaded these data sets. I can also, there's also the option to share the history with you guys. So I think I should do that. That will be an easy option. I'm again pasting the links in the chat box, so you can do this by both the options. Either you can upload all those data sets, or you can use this particular option. So when you access the history, you'll be greeted by this page. So I think a few things are still running as of now, but I'll keep on explaining this. I've also added one GTF file. It was a big file, so need it per se necessarily for this particular analysis. But when you do your own data sets, your own files, then do use the annotation files. After uploading the data sets, the first thing I think in the morning, Prashant also explained, first thing we do is we check for the quality. So we do this by using the fast QC. The steps are quite similar for command line in Galaxy. In the command line, it's just that you are greeted with this kind of interface, which is not very user friendly for people who are new to RNA-seq or these kind of analysis. So Galaxy is a good gateway to understand certain things, and with using Galaxy, a lot of experimentation can be easily done because all of the options, they are very user friendly. So here, after you upload the data sets, they will be shown like this. So in that, we need to, for the fast QC, there are multiple options. If I just want to do QC on a single file, single data set, if I want to do on multiple data set, this for this, if since I have multiple data sets here, six in total 6 files are there, so I'll use all these files, so 6 files, and with both reverse and forward reads. that gives me a total of 12 files to perform the fast QC. So I just need to select all these files, fast QC analysis, all the files. Normally the fast QC is run very fast, but I don't know, today, since we are accessing Galaxy from this particular IP a lot, it's maybe not responding very well, but we'll still try to get the results. So after selecting the files, you just, if you have the contaminant lists, we can add that. The adapter list, usually the kind of the sequencing platform that is used uses a particular type of adapters. So if my sequence was, if my upon Illumina 200 MySec or HiSec. So for in the library preparation, a particular kind of adapters would be used, so which would be present in your sequences. So all those lists are available online freely with the companies provide these lists. You can also upload those, but we are going to ignore this as of now. Then there are various other options for reads greater than 50 base pair. I'm selecting no because I don't want to group these. A lot of explanations are already given. The beauty with using this Galaxy server is that when you scroll down, you will get a lot of things, what the tool does, how it does it. So for example, if I scroll down towards the fast QC here, so it will list the purpose, it will list how, what kind of this thing, inputs it is going to take, the input and output formats, what all things it is going to perform. I think these have already been explained in the morning, so I'll just execute this. My job will be listed somewhere on the top. I think it's still sending. Yeah. So for my 12 inputs, I'm getting 24 outputs. So outputs is quite a lot, but we don't have to worry because the raw data will be in form of graphs in the web page. So I have already done the QC for these files. I'll just show you. These are the same samples that I have been working on. I just started this analysis in the morning. Should be finished by now, but for some reason, CuffDiff is taking a lot of time. But anyways, if I click on this icon, view data icon, it's a very small icon. If I click on this, I can be able to see my raw data. So these are all the statistical analysis that is done by FastQC. It has read a lot of things in the sequences and produced all these results. But for me, these results, I am unable to interpret them very well because all of this is presented in the form of data. So what I would rather prefer is some sort of a graph or some sort of a pictorial representation. So if you click on the FastQC data web page icon, so you will be presented with the HTML file. We have all the statistics or all the summary that FastQC report produces. The basic statistics says that it was done on Sanger or Illumina 1.9. Then the sequence length was 154 with the GC percentage of 54. This particular picture, it shows the quality of the reads. If I'm getting a score below 20, it will be very bad. If my scores are in the green region, then I would say the quality of the reads is quite good. So for this example, the read quality I would say is pretty average. So a lot of statistics and lot of data is presented, which I think still was all this Prashant explained it in the morning, right? So I'll just move on to the next part. After the pipeline, our next part would be doing the alignment, the indexing and the alignment. So on Galaxy, we don't need to index the human genome because we can use the inbuilt genome in this. For alignment, For RNA-seq, usually we prefer this HiSet 2 instead of Bauti 2, because in our lab, using these tools over the time, we have found that HiSet 2 provide better result for the data. So I'll just use HiSet 2. So it says a lot of details about what is HiSet. If you are new, you guys can read all these things. You must read all these things to understand how the tool works and what all options can be used. All these options can also be used over the command line as well. The options remain the same, the tools remain the same, just the interface is changing. So here I have to either I can use a genome from my history or I can use an inbuilt genome. I'll be using an inbuilt genome, or I have the option of using only the small genome, that is the part of the sequence chromosome what I have downloaded. Already done that. Whatever organism you want to use, so Homo sapien. Using the HG38 build instead of HG19. So I'll select HG38. The library I checked in the website, the data provider, the libraries were paired end. So I'll be using the paired end. Now here again I have the option to use single data set or multiple data set, or even I can use data collection if I would have provided that option during uploading my data. Multiple data sets for this option as well. For this, the fast queue number one is the first file. That is the forward read, and here it is the reverse read. Read, I'll select the files with the R1. I would also just suggest you guys to label your data here in the history itself, so it's a little easier to find out. Because if you keep on uploading a lot of data in Galaxy, it will you are going to get confused. So labeling the data can be done. So you can edit the tags here as well by clicking on this small icon. When you click on this small icon, another icon. So this is replicate third for the reverse read. So I can say this is normal. I need to edit the tag. It doesn't allow spaces actually. So I'll just edit the tab. Instead of space, I'll use a underscore. So normal underscore duplicate 3, and it's the reverse read. If you guys, I don't need that right now. It's sometimes useful. So moving on, I'll show you the HiSet 2 aligner. So in the HiSet 2 aligner, we have selected the inbuilt genome as of now. We have selected our reference as HG38. We have selected that it is a paired end library. Now, in the FASTQ file number 1, this option, I have selected multiple data sets, and now I will only select my forward reads. So my forward reads will be labeled as R1 at the end. So I think total of 6, because there were 3 replicates. Total of 6 files are there for the R1, and then for the R2 I need to do the same. Again 6 files for this strand information. If you are not very sure that what it means, you can leave it to default. I'm leaving it unstranded because I couldn't find this information, but usually it's provided by the service provider. Kind of the machine that we can use. There are again many advanced options that can be used. If you are using Galaxy, I think you should stick to the default options for now. But as you get comfortable with experimenting on the dataset, you can tinker with the various options. So after doing this, we just need to click execute. Shows me the output as output that it has executed the HiSet 2 aligner and had added 6 jobs. So I had added 6 jobs. So in these 6 jobs, forward and reverse reads. So total 6 files were there. So that makes when I align those forward and reverse reads, so it will give me a total of 6 outputs. So here I have gotten 6 outputs, which is correct. So these outputs are already in the BAM format. BAM is nothing but binary alignment mapping. There are 2 types of mapping files, BAM and as well as SAM. So SAM is sequence alignment mapping. SAM is human readable, but BAM is binary. So it is easier for the computers or the computers to read and process. It saves a lot of memory and it increases the processing power. In our pipeline, we need to convert the SAM files to the BAM files. That is an additional step. In Galaxy, it does this by default. Taking So after doing this, we'll be getting our data as the output. I'm just going to show you the aligned reads. Yeah. This example, and click on the view icon. So your file will look something like this data. So as you can see, these are the header files starting with the @. The tags are all on the top. So the queue name, flag, R name, position, map quality, cigar score, that data we can find after the headers. A lot of headers will be produced. It takes a little while to load the file. So here is our aligned file. So you can check those as per the yeah. So here I would also like to show you the statistics that you should always check when aligning the files. So a total of this many number of reads were present in this file. 100% were paired. So we selected the correct option that these were paired and reads. So now out of 3,90,000 reads, this is 95,000 reads were not aligned at all. So that is 24.3%. Then 70% reads were aligned exactly one times with a reference, and then around 6% reads they were aligned more than one times. So we need to check these statistics. If we are getting the alignment as lower level of alignment or lower percentage of alignment, we need to tinker with some options, or either some of the options that we have selected is not correct, that whether our data set might not be paired and it might not have it might not be forward standard or reverse standard as those earlier options suggested. So this is the output we get after running HiSet 2. We need to it's as simple, we need to sort the files. So for this we need yeah, samtools sort. So I'll just type sort. I think this will get me the result. I'm unable to see that. I'll just type samtools. I think I lost that option somewhere. It was, I just checked that in the morning, it was here. I'm sorry. I need to scroll down a little bit. Yeah, okay. I'll just use sort SAM instead of samtools. It also uses the same thing. We can use the beauty of using Galaxy is that there are multiple tools available. Uh, I prefer samtools because we have been using that for a while now. But yeah, samtools sort. Here it is. Now it's visible. I don't know, for some reason I was unable to see that. on SAM tool sort, we'll be greeted with this kind of page. So we need our aligned BAM files as input. So I'll do the multiple input this thing. For multiple input now, I'll use my aligned BAM files, right? So I generated those in the morning, or we can use, these are the jobs 353 to 348 to 353 that I just did. I'll select these should be 6 in number. These are correct. I'll sort them by coordinates so that the chromosome 1 will be on top. But here in the sorting, the computer doesn't know. For computer, after 1, the chromosome 10 will come, and after chromosome 10, the chromosome 11 will come. So this is how the computers sort. Instead of doing it the human way, humans will generally sort. If I give you a bunch of numbers between 1 to 10 and jumble them up, 1 to 22, and jumble them up, you will go 1 2 3 4 5 6 7 8 9 10. But computers don't do like that. They'll just see for sorting, they'll first go for the chromosome number 1, then they will go for chromosome number 10 because they see a 1 in the beginning. Then they'll go for the chromosome 11, then 12, 13, 14, 15, and similarly they'll do that. So after I'll select with the coordinate. What sorting here actually does is it helps us increase the processing a little bit. We can also generate indexes that can be visible later. So it's using the sort option is as simple as that, selecting your data set and clicking execute. So again, I'm greeted with the same page. So all these BAM files, I think I have selected one extra file, selected only 6. So yeah, earlier from 96 was my earlier job, so I selected that as well. Anyways, but these 7 files, or I should say 6 files, should produce 6 outputs. These outputs are nothing but the BAM files which are in sorted this thing. Are you guys able to follow me until this point, or do I need to slow down a bit, or anything you want to ask?
I think it's excellent. One I am. I think we should also give them enough room, to do the exercises. So maybe once they do until cuff diff or something, cufflinks something, maybe we can ask them next. That was what I thought. So are you done with this, Sam, or you want to give them another round?
I think I can just finish up till cuff diff on Galaxy. Then I'll just go through the options, if that is okay.
Yeah. Yeah. No, command line we can probably even give it tomorrow as well. I am okay. So just
okay,
give them. Yeah, I think we should give them. The demo until cuff diff, and then we should give them at least 1 hour. Today to do the exercises, and in between probably Ashwini will give another demo on DSync. Ashwini, and yeah. Thank you.
Yeah, so I'll just wrap this up in 10 minutes. Then I think there will be plenty of time for the candidates to do their analysis on their computers.
Yeah.
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 Okay. So after doing this, we just need to cufflinks. So cufflinks we use for generating the assemblies. I'll search for cufflinks. I'm a little, I don't want to scroll a lot, so I type all the options here and they appear. So that is amazing with Galaxy. So I'll click on cufflinks. Again, I'll select all my data sets. I'll go with multiple data set option instead of a single file. I'll just go for the multiple data set. Or maybe I think I should go for a single data this time because I have already processed all those things. So here, the thing that anybody need to remember is that we should give the sorted files here. So as you can see, the SAM tool sort on data 350. Sorry. Yeah. Yeah, Ashok, we'll take a break in 10 minutes. It's almost over. So I just saw your message. We'll take a break in 10 minutes. It's almost over. Then you guys can take a break. So I'll just select one data set here. So I'll use SAMTools sort on data number, my job 360. So here we can select the maximum intron length. Here, if we want to, now the Galaxy is still explaining us a lot of things. So if I use the maximum intron length, then the alignment with gaps will be ignored. So I will, as of now, I will use it for the default option, that is 300,000. Then the minimum isoform fraction, this suppresses the transcripts below a certain abundance level. What this means is that if a particular transcript is below a certain level, that is 0.01 per abundance level, what cuff links will do is it will just discard that particular sequence. If it keeps on finding those particular sequences, then the isoform fraction will increase, and if it hits the threshold value of 0.01, if it is above that, so that will be considered. The same goes for the pre-mRNA fraction. So all these options are already pre-built. Slider. Usually for the first run on a new data set, what I do is I just I just anything. But as later, if I get some solid results, then I start filtering. These can be used as some sort of filters as well. So here I want to use a reference data set. So I'll use my reference data set. The thing is that I already uploaded that reference data set a while ago. So this is available on GeneCode. The file is quite big. If you want, I can just paste the links for the human. This is version 25, I think, what I have uploaded. I'll just check that. Here, you guys can directly get, I'll just paste that in the chat. So I got my reference from the GeneCode itself for this particular project. I have selected my reference. Now I will count hits compatibility with reference RNA only. The option is explained quite well. I'll just move on and select yes, because that is what I thought was good for this particular data set. Then I also used multi-read correct. All these options, length correction, I just use the cufflinks default, and then I executed the job. This takes us far. I think one of my jobs failed, but so this is how you see if one of your job fails. Command line option, the troubleshooting part is a little difficult, but it can be done. In Galaxy, you get all these color codes. So all the green jobs, they have been executed, they have been finished. The yellow ones are still processing. The red ones have failed. So I might have messed up some of the options because I've been running with a lot of combination and permutations for this particular data set. I didn't have a lot of details for this. I've just been testing that as of now. One of the job has failed. I can just delete this, and it will be hidden from my history as of now. To look pretty, okay? I don't like the red
just a
failed
comment.
job.
Just a comment. So what I am has taken is only one small file, with about one chromosome or two chromosomes,
One
just
chromosome,
to give
yeah.
to give a demo. So usually, you would really expect that. So if you have a big file of yours, you can definitely analyze and you can do that. So since this is a kind of a demo, just to save the bandwidth and save the time and energy, this is a kind of a simplest demonstration. Okay. Yeah. Thank you.
Yeah. Thank you, Prashant. So this leaves this leaves us with the last option of cuff diff. So this will do the differentially expressed gene analysis for us. So I'll just type cuff diff and click on the option here. So here in between, since I'm using replicates, I also need to use the cuff merge option. Will do is that cuff merge. Yeah. So what cuff merge will do, since I am generating six different assemblies for this particular data set. So I had given it earlier I had given it 6 inputs. I'll show you those inputs. So cufflinks, yeah. So these cufflinks, in the cufflinks inputs, I got the assemblies in this forms. So it will generate a total of that will be comprised of skip transcripts, whatever transcripts it wasn't able to find, then the total map mass, then the assembled transcripts. This is the file that I want to use further. Then transcript expression, then the gene expression. So I would like to, I would use the assembled transcripts for this particular cuff merge step. So what I would do, I've already performed that particular thing. So for transcripts, I'll use the I have used the multiple option. I'll select all my assembled transcripts here. So you need to keep button pressed to select. If anybody missed that, it will be a little finicky. You might think that your system is not working quite well, but that won't be the case. You just need to press the control button to select the various these things. So once you select, you are not going to be able to deselect it just by clicking outside. So don't worry about that. So we'll use the assembled transcript file here for the cuff merge. The cuff merge the current version, the Galaxy that is hosting is 2.2.16 is not working for me as of now. So I switched to 2.1.5. So now it has switched. So it is showing me that currently I'm using the older version. I'll select all of my assembled transcripts. I will not use my skip transcripts in merging the assemblies. So that won't be any use to me. So I'll just use that option. Then I'll go to select the data type. Since I was using this data set, I had two normal and there was the tumor. For the normal samples, the files were, for me, they were from 96 to 98. And for the tumor, I need to select the other jobs. So 99 to 101. So I'll use those. If I had more conditions, just like in RNA-seq, there can be varied conditions with various data sets. There can be a timestamp as well. So all those conditions can be incorporated within this thing. If I'm doing research on some planned data set, there might be the various conditions might be the height, the color of the, if I'm talking about just the flower, so can be the color of the flower or wherever the plant was kept or what all different conditions that have been treated, the plant has been treated with. So I can use all those options. The same goes with the humans. I just use the plant option because I find that easier to imagine. I'm sorry. So I'll just move on to the next part. So I'll use my classic FPKM analysis for these samples because I found the results better. For the dispersion method, the Galaxy says that if I'm using only one sample per condition, then I'll go for the blind dispersion method. But I can go for the blind as well, but here we are using replicates in this particular option, so it doesn't matter. You need to read about all these methods because the backend algorithm does a lot of things which any bioinformatician or the person handling the dataset should know. at least read a little about it so you get a hang of it. Use the I'll skip all these options. So bias correction, I would like to do that. And from this, I'll use the reference genome. After that, uh, I'll skip this, and I'll execute. Was the cuff merge part. Now, after running the cuff merge, we will be getting one file, that is the merge transcript. I think I'm over time, but I will take another 5 minutes. So here, when I gave this all these inputs, I think I messed up a little of some input. I'll just check that. I'm sorry. I think I missed one of the files. Since this is a demo, if you guys are facing any problems with your datasets, please do let me know. So after that, we'll get the merge transcript file, which will be looking something like this. Have the annotation of the whole file is annotated. Since we use the annotation file, I'm able to see my whatever information was given in the gene code file in this file, along with the uh, dataset, the data which was provided in my dataset. So both those files have been merged, so I can process these further. I'll use the last option, that is the cuff diff. So for cuff diff, here we need to use our assembly. As I have labeled that as merge assembly, so I'll use that. So here I'll use the input. We can have this thing, input that we want to give. I can give a list of single replicate conditions. So the list of conditions I need to provide, or multiple replicate conditions. So no data set, haven't uploaded any of those data sets. So I'll use the SAM and BAM option. So here I'll again name my conditions name normal. So in the normal, I have prepared. So for normal, it was these three files, and then for tumor, I think 100 and 101. So again, I'll use the same options. I'll go for the blind one. The FDR is the false discovery rate. For statistically correct, we use FDR. So what FDR does is it helps us with regards of the multiple hypothesis. So when we are using multiple samples, so uh, the p-values that we obtain can be false or can be false positives. Overcome that, FDR or Q values are being used. So I can set the threshold. 0.05 is pretty good. If I want to be very stringent, I'll go for 0.01. But I think I'll go for the 0.05. So I'll go for the multi-read correct. Then I'll again perform bias connection. Correction will use the HG38 reference. It is already built into Galaxy. All these options on the command line, we need to use those options manually. So now the only thing left is to execute the job. So a lot of jobs have been queued. I think see, a lot of jobs have already been queued. So again, I have selected 8 inputs for this. So okay, I again clicked on 353, which I didn't want to process. So anyways, there should be 7 inputs. The 7 inputs should be the sorted files. These are the 6 sorted files, and then the transcript assembly. And then this produces a lot of these results. So transcript FPKM, gene FPKM, the gene differential expression testing. This is the file that we are interested in most. Then the the differentially expressed promoters are also being given to us by the CovDiff program. So I think I would so that he can guide us further in the workshop.
Yeah, thank you so much, I am for the excellent presentation and demo. That's a wonderful one. So if you have any quick questions, so please, all of you ask I am. I'll be there until 6:15, and you have to do all the exercises, please. Is Ash here or Karan? Would you like to give a quick demo for 5 or 10 minutes, or maybe you want to do it tomorrow if you have time? Yeah, Karan? Yeah.
Yes, sir. Hello.
Yeah, yeah. So Karan, would you, is the server accessible to you? Or,
Yeah, I can. Just give me
okay.
2 minutes. I will.
Yeah, please, yeah. So please start sharing your screen, and after this, friends, Karan will take about 5 to 10 minutes, and then you're done with it for today. But please don't go away. Ayam will be there, Shalini will be there. I think you all have to do these exercises, okay? I think in fact, when you are not there, we thought of splitting you into different groups so that we could have those exercises done, okay? And tomorrow, of course, you have plenty room for exercise as well, but you have to do these exercises, please. Huh? So if you have any questions, please let us know. Yeah. So Ayam, you may stop sharing the screen so that Karan can share the screen and do that. Yeah. So any questions, please, all of you? Yeah. I know you have to do exercises. It's very important. So please feel free to do those exercises. Any questions? Please feel free to know. I can see two Northeast researchers. In fact, three. In fact, Imakshi is also there. Of course, she's there. I think Deeptan Laskar and Kikusenyo Kyuho. Any questions, please? Yeah. Yeah. Any questions, Ambarish, Anchita, Ankita, Arunima?
Would you introduce the dataset again? I would like to ask. Hello, it's question from Ambarish side.
Yeah, Ambarish, yeah, please, yeah.
Yeah. Would you introduce the dataset which are used for the CuffDiff and CuffMers?
Yeah. Maybe Ayam will share. If you type your email address, yeah.
Okay, sir. I just text the email address.
Yeah, please. Ayam, I think would you mind sharing those data sets to Ambarish and others, whoever is asking? Yeah. Yeah. So any questions? Ashokji, Esther, Gaurang, Jyoti Sharma, Karan Patel, Kemraj Thakur, Yasin, Sabul, Madhumita, Nandini, Neha, Nimisha, Nisha, Rajul, Rashmi, Rosaline. 2 minutes, yeah, please. If there are no questions, yeah, Karan, please just give a quick demo of 5 minutes, and then they will do the exercises. Thank you so much. Yeah.
Yes, okay. Hello. Hello.
Yeah, Karan, it's audible, and your screen is also visible. Please.
You can see the Linux part.
Yeah, very much. Yeah.
So I have the data. I have a count table in this folder, a workshop folder. So I want to do the PCA analysis or differential analysis. So in R first we have to import the library DSIG2. And then some table manipulation library. After that, I will import my count table that I've shown before. This is how my count table is looking like. The first column is genes, and then the count replicates counts, unaffected versus infected. So I have to remove the all the rows, all the columns, all the rows that contains all the columns with zeros, so that biasness can be removed. So the we first calculate the row sum of the each and every rows, and then we filter out the row sum that contains less greater than equal to 0. And then we make a data frame. So this is how my expression data set look like. We have filtered out, if we see the dimension of count table and the exp data, there is a decrease, there is decrease in rows. So this means that we have filtered out the rows that contains all the rows that contain zeros. And then we have to make the metadata according to our samples. So in this, the data frame and column names of experimental data, that is UNF1, 2, 3, and INF1, 2, 3. And what is this? A condition means what is the sample? Means control or infected. This is my metadata of the table, and then we will check that all the row names of condition is contains in the column names of experimental data. This shows that means row names of condition is present in column names of exp. And then we change the exp row names, count row name according to the metadata. And then we once again check for double check. We once again check that row names and column names are equal or not. It's true. Then we make a DSIG matrix in which we calculate the we calculate the differential test. Then we find out the result between the groups. this is a table from the differential test. And then we want to plot the MA. So we will give plot MA RES main is equal to MA plot. This is the MA plot we have got from this. And then volcano plot, which is between log to fold change and the PIDJ values. This is how our volcano plot looks like. The red one is significant one, and in log to n x-axis, the minus ones are down regulated ones, and the positive ones are up regulated ones. Then we scale our logarithmic scaling is performed. And then we plot our wait a minute. And then we take the PCA data, calculate the PCA data. This PCA data. In this, the PC1 and PC2 columns are important, which contain scattering points. And then we calculate the percentage variation in the PCA. And then we plot it. And also label the plots. So UNF1, UNF2, and UNF3 are seen to be clustering, and INF1, 2, and 3 are likely to cluster. So PC1, so between the UNF and INF, the variance between these two are 68%, and the PC2 is showing 15% variation between intra sample variation. Thank you, sir.
Yeah, thank you. Thank you very much, Karan, for your time and energy on this. This is awesome. So it was wonderful session from Karan, Ash, and I am demonstrated this. Now it's time, we proved ourselves and I've done the exercises. So I know it's very difficult to do the exercises in R and using the DSeq, but it's time, you did it using Galaxy Quiz. So we are here, and the session will be open until 6:15 p.m. So I am will there and myself will be there. I've got some other meeting I might leave, but I am Shalini are already here. So thank you so much. So if there are no questions, I think we should thank Karan and Ash for their time and energy and spending it in their wonderful time. Yes, Saurabh, please, yeah.
Can we have a summarized list of all the commands Karan has used just now?
Yeah, I can give you the HTML file with all the commands and all.
Okay.
Okay, I will in the message box. I will paste it here.
Okay. Yeah. Thank you. Thank you.
Yeah. That would be good. Yeah. So thank you so much. So if you have no questions, I think we should probably stop here. Okay. Yeah. So please do your exercises, please. This is very important. So we are here now until 6:15 at least. So I got at least 1.5 hours, at least to 6:30. So please feel free to ask questions. Yeah. So thank you so much, Karan. Thank you, Ayam and Ash. Thank you so much.
Thank you, sir.